I think that's just like good general housekeeping just to help the conversation progress forward. But, obviously I think that Jace has some integrity and I don't think we're gonna have a problem either way.
All
Yeah, I'll
right.
do my best
Well,
to keep it smooth.
Oh yeah, and by
so
the way, is it
Fine.
Jace or is it Jace?
no one knows. It's a mystery for the ages.
Okay.
Yeah,
All right.
that's funny. You
Yeah.
can just call me Jay. It's fine.
Jay?
Yeah, yeah.
But I'm just curious, is it pronounced Jace or Jace, like for your name?
Should I reveal the secret? It's
Yes.
it's supposed to be Jayace. So
Okay, I'll just call you Jay then. I might still call you Jace, I don't know, but we'll see.
yeah, a lot of people say like JC, or some people say Jays. You get it right. But a lot of people say JC, but you just say Jay is fine.
Okay. Well, for now, I think that we should get started if that's okay.
Yeah, yeah.
I think is everybody up here? Is all the mods and everything up? Are we good?
Yeah.
I only have me as a speaker. I actually don't see anybody else. It says speakers 4, but then it only shows me. So it's kind of confusing.
You have like a drop down menu on the
Yeah, I did the
right-hand
drop
side.
down menu and I only see me.
Strange. Well, right
I
now it shows
see.
Let me
me,
try to
Geo,
come back
you,
in
and
time.
Jay.
Let me
Yeah,
drop
I
down.
see 4 people.
I get to invite her back up. One sec.
Okay. We're good.
Yeah, it should be all good. Everything's set on this end. So yeah, if you guys want to share screens and stuff, you can up here too. So I don't know if you want to, but yeah, you can do that.
Cool.
Geo, does it look better on the live stream if we drop off the stage, until they need us or whatever? Is that better for the live stream?
Let me see. Well, if they're not sharing their cameras, I guess maybe, yeah, I guess so. It'd be a little bit cleaner.
I don't think we're gonna be Jace, do you expect to share? I might share like papers. I don't know because I don't have good bandwidth right now. I'm actually like using like a hotspot on my phone. So like I don't know if I'm gonna be able to do screen sharing. Do you plan on doing screen sharing?
No, we don't have to screen share or do any kind of video sharing because I'm out
Okay.
walking around right now. So
Oh, we're good then. Okay. All right. Are we good to go, Gio?
Yeah, I think everything's good.
Okay.
I think Galatrax and I will just jump off the stage just so it's only your two avatars or whatever, just for the video. But we can pop on real quick. We can still mod even if we're not on the stage. But yeah, no, you guys are good to go. And yeah, just let us know if you need anything. But should be good to go. And then is there going to be a question and answer after? Or
Yeah. So I would
do
need
that?
somebody up here at that point to like read out the questions. That'd be very helpful.
Yeah.
But I already
sounds
know
good.
Galatrax gonna be taking note of them. So just I guess at the end, like maybe he could come back and help and read them out and stuff. That'd be good.
All right.
Anyway, just whatever y'all want to do.
Yeah, I think you two are good to start. However you want to do it. If you want to like do opening statements or opening positions.
Yeah, I think that we should give like an introduction, like to why, to why we're interested in this topic, what research we've done in it, what's our area of research, just general things like that. Do you want me to go first, Jace, or do you want to go?
doesn't matter. You can go first.
Okay. So basically, I study biology, particularly cell biology. And I've always been really interested in the origin of life research. I've written some papers about it in undergrad, and I am still interested in it. And I have looked into like the scientific, the scientific studies of it, the origin of life research, and like where we're currently at in the field of origin of life research. And I've come to the conclusion that I don't think it's like a feasible hypothesis, right? I don't think it's been demonstrated to any level that would give the theory of origin of life, accidental origin of life, any like feasibility. So anyway, yeah, and then I was looking for some debate opponents, and I was told about Mr. Jayace right here. And he's, you know, talked about it in the Discord channel a lot, and I saw that he's posted a lot of papers on it, so I was really excited to talk to him about it. And I've been looking forward to this conversation. So yeah, you can give a little intro if you like, Jace.
Okay, well, thank you. I appreciate that. But unfortunately, I will say, if you're looking for someone to do like formal like debates and stuff with consistently, I'm just gonna be unreliable for that. I'm just too busy. But if if sometimes we can schedule discussions like this, or if you happen to, I just pop in occasionally, usually, and if you happen to catch me when I'm there, that's the best time to get me. But nonetheless, I am Jayace, or Jace, or just Jay. I'm also studied biology, but also chemistry, aka biochemistry and molecular biology. Uh, and I currently, well, I was doing research in the pharmaceutical industry for a bit, but right now I'm actually helping a colleague do his PhD in something SC mentioned earlier, looking for DNA in dinosaur fossils. But yeah, my position on the origin of life, I've done some undergrad papers or whatever, lab work and stuff, but I haven't done anything like professional on it. But my just position is kind of the opposite, and I think it's quite a vast field, and it demonstrates quite a plausibility of it's not really accidental nor random. It's just how the pattern of emergence works along with the laws of thermodynamics, where you get more complex and complex chemical systems because they can disperse entropy the best, and it just ends up in something akin to life, if not life, just naturally. It's just how nature works. That's pretty
Mm-hmm.
much my position, and that the people doing the research in the field demonstrate that quite vastly in a multitude of ways. Where I've kind of expertise on one particular area, which would be obviously the biomolecules. Like I have understanding of how those form, both in lab conditions, in biotic conditions or living conditions, and in abiotic conditions.
Mm-hmm.
So that's like
Can I
my
ask you a question
extent
about?
of understanding.
Okay, cool. I want to ask you a side question. You said you're doing dinosaur research. Is this like for evolution?
Well, my colleague is doing his PhD in molecular biology, more like molecular genetics, kind of, I guess, would be the field. But the project that he undertook was what he's doing a evolutionary hypothesis on genetic evidence linking dinosaurs and birds, linking the clades together through molecular genetic evidence. So we got all these different fossils shipped to us, these different dinosaur bones shipped to us, and we're doing certain chemical processes to try to extract DNA or RNA, but we want to find DNA, and enough to actually sequence like an actual genome, or at least a large portion of the genome, so that we can actually compare it, do phylogenetic comparisons with
Cool.
modern bird genomes. So.
Are, are you co-authoring with him on that paper? Are y'all working in the lab together essentially?
Yeah, I'd be a co-author. He'd be the lead author, and I would be the co-author on this, probably. Me
Oh,
and a
cool.
few other people would co-author, but I help him right now. It's pretty much every day, because we're just doing the screening process. Like, we have thousands upon thousands of little specimens to go through, so he has the screening process, this chemical process that we do to screen. And so we've been doing that for like the last month or two now, pretty
Is this
every
like in a
day.
university setting? Or like a
Yeah,
there's
yeah,
like
yeah.
oh cool. And you've already got your PhD, right? Or you're in graduate school or just
Yeah, I'm still in grad school too.
oh cool. Awesome. Well, I feel like we're at the same level of education. And so this would be like a cool combo, like and it's awesome that you're like doing research on evolution and stuff. That's super cool.
Well, thanks. Appreciate it. Yeah, my professor just my like PhD project probably get back to working on it for a while. We had the COVID stuff, so I was kind of either helping with that or just kind of in limbo while my professor, my PI, was helping. But should be back on track now here shortly once I finish helping with this dinosaur thing, because that's if we were to find you know, DNA, enough DNA. Even if we just found any DNA, that would be a pretty significant, potentially like Nobel Prize winning thing. But if we found enough to be able to sequence to compare to a modern bird genome, that would be just totally game changing for
Have
dinosaur
they
and
the sequence like
bird.
dinosaur DNA yet?
No, the
Oh,
dinosaur
really? I thought that
genome.
they sequenced from the
No.
I thought that they have sequenced dinosaur DNA. Okay, wow, that would be revolutionary, dude. That's awesome.
Yeah. No, nothing from that long ago has been sequenced. Maybe a few rare animals that were preserved in like very specific ways, but no dinosaurs that I know of from
And it is soft tissue,
that
right?
far back. Say again?
Is it soft tissue that you're working with?
Well, we're looking to extract soft tissue from the original bones that we get, or from the fossils that we get. We're looking to extract soft tissue out, and then from any soft tissue that we can find, we're looking for DNA in that. You have to first separate the soft tissue, the stuff that would contain cells, from the mineral and the mineralized rock that the bones or the fossils found in, and then you can do further breakdown to look for actual genetic material in the soft tissue if you find any. So we found some like remnant things so far here and there that we need to like, that are like preliminary positives, but they're preliminary positive. So there's still a lot of testing that we have to do, but they get to move forward past the screening process. Probably have like 10 or so specimens so far that have came up positive on the screening process. So, but could be due to error, could be a flaw in our procedure. So it's, like I said, it's just a preliminary positive so far that we
Yeah,
got.
that's really cool. I've heard about the soft tissue in dinosaur bones and basically the case that like if they were millions of years old, how would we still have soft tissue? But that's a different conversation that we don't have to get into, but that's really interesting.
Yeah. That's more of a paleontology, evolutionary kind of conversation. But yeah. Well, that's my story, basically. That's what I do basically during the week, sometimes on the weekend. Sometimes I get the weekend off, like today.
Cool, that's awesome. Yeah, maybe like in another day we could talk about the soft tissue and the dinosaurs because basically the rebuttal to that was that oh it was just like some bacteria and stuff and it wasn't actually the dinosaur cells themselves. But if you're trying to get DNA from the dinosaur cells and the dinosaur cells are soft, then that would definitely be a rebuttal to that claim.
Yeah. I mean, people, I guess it's already pretty accepted that there is like at least remnants of soft tissue uh, that's been found by people like Mary Schweitzer and Mark Armitage, or Armitage, I think his name was. Um, like those are two basically different people that may have found at least remnants of soft tissue. And so chemists have began working on and biochemists have started working on preservation mechanisms for, because it was just kind of assumed that genetic material, or that at least the soft tissue, wouldn't last for millions and millions of years, but no one ever formally showed that mathematically, these tissues and cells and genetic material and all this can't last that long, right? So it was just assumed. So it leaves the possibility that under certain conditions you can actually get the chemical conditions to favor the preservation of these
Have y'all carbon
materials.
dated that soft tissue? Not that carbon dating's accurate, but I'm just curious.
Um, you don't carbon date it because it's just too far back. We do uranium, lead, argon, fission track, thermoluminescence, those kind of dating methods we have done, particularly on the preliminary positive samples. We got to know where they're from exactly. We have the coordinates and stuff. We have the ages from all these different dating methods, and they all converge on the same age for the specimens. But that's all part of the process.
Carbon dating might be useful too, because if you're just assuming that it's that old, I mean, it still would be worthwhile to carbon date it, just confirm that, I
Well,
don't know.
some of the stuff does get carbon dated. Some of the material that the specimens are found in will be carbon dated, just depending on which strata that the fossil was found in. So you don't want to put the fossil through that because it'll get kind of destroyed in that process, but you can put the strata that it was contained in, whether it's a fossil or bone, whether it's rock or bone, mineral or not. You can date the strata using carbon dating. But then you also, right, I can already tell you for the 10 or so samples that we have, you can't make out the half-life has gone so low. The background carbon-14 radiation is the signal from that background, the noise is far larger than any carbon-14 signal that we can find in the sample. So that what that tells you is that it's much too old. You can't distinguish a signal from the noise. So
I would like to see some papers carbon dating dinosaur soft tissue. That'd be really cool. If they had any, that'd be interesting. Because from what I understood, in terms of dating the strata, they would date the strata based on the organisms that were found within it, and then they'd kind of right? No,
no. We're talking
no.
about absolute dating. So you
Okay, absolutely.
carbon
Okay.
dating and
But
radiometric
wouldn't you need a
dating
reference point for carbon
is an
dating?
absolute dating method, where the type you're talking about, where you use the fossils to date the rocks, or the rocks to date the fossils, that's just relative dating. Sometimes it'll be done in the field. If you find a fossil and you just want a quick know of its relative age, you use the strata. If you know the age of the strata, then you can use it to date the age of the fossil. But like I said, those are
Okay.
relative.
Yeah,
Carbon
I
and
think this.
radiometric dating is absolute.
Yeah, I heard you. So then I wanted to say, for the carbon-14 dating methods of the organisms, wouldn't you need to compare it to something of a known age? Don't you have to have comparisons for carbon-14 dating? You have to say, okay, we know this is this age, and so we're trying to find we're trying to find out how old this other thing is. So we're gonna carbon date it and then compare it to something of a known age.
Yes, we have a reference sample at our, because we
So
use
then, how would we age the
ACMS.
reference?
What do you mean? These are absolute ages, right? It tells you exactly how long ago the cutoff point or the formation point for certain isotopes are, right? There's no leniency or guessing, right? It just tells you a date of how long ago that it was
Are you
either
saying that
formed or that cutoff point is, right? So we can date if we need a reference for carbon dating, and these papers have been published of things that people have witnessed forming, rocks or organisms that the death was witnessed or whatever. So the cutoff points are witnessed by the scientists themselves and then dated using carbon dating methods to be 0 years old, or however, if it's Mount Vesuvius, it'll be however many years that, right? So you get the dates for those things that have been witnessed, and now those can be used as references. So you have an absolute reference that you can use as a control when you're now dating your new sample that you want to date.
Mhm.
If you want to make sure that your mass spectrometer is working properly in the radiocarbon dating sense, you have a sample that can be used as a control. So if the 5 other labs that have done the QC on this sample have gotten 35 years and you get 22 years, you know that it's probably not all of them whose instrument isn't working right. It's probably your instrument that's not working right. So then you can start to look for either your procedural error or your error in the instrument, right? So
Mhm.
that's how a reference is formed.
Right. And so, like, how would you get like the accurate references for things like millions of years ago?
There's there's like zircons and things that can be used in mineral samples, like strata samples that are taken for certain different data radiometric dating processes. But like I said, for some of these, like because it's an absolute method, you don't need an outside reference, right? There are some available, like I said, zircons are one example of figuring out the age to use as a reference, but you don't need that necessarily for radiometric dating because it's absolute.
So uranium dating is the one that I remember you saying, and I looked it up, and it said it does need a reference when you do dating like uranium, using uranium. Is there
Yeah,
another?
there's minerals that form in the modern day that form with uranium that can be used as a reference, and there's also helium kind of
Mhm.
depletion methods that can be used to reference samples for uranium, lead, and also argon. The same thing. Certain minerals form in the modern day with like argon leakage rates, and you can figure out references from that. But again, you don't really need all of this. All this reference that you're talking about is just to calibrate your mass spectrometer, right? To make sure that it's not off, that it's not giving you the wrong values. So you ideally you can just use something that can be radiocarbon dated if you wanted to do a reference like we were talking about earlier with organisms from Mount Vesuvius or modern day organisms that you witness the cutoff point, right? Because you're just calibrating, you're just using the reference to calibrate the mass spectrometer. That's all you're really using it for.
Mhm. So basically, like we're gonna be looking at things like, for example, with carbon dating, just like for the audience, in terms of like dating some of these organisms. So like carbon-14 dating, you're gonna be looking for the amount of carbon-14 levels over time. And then so you're basically gonna be comparing your unknown sample to a sample that has a known age, and you're gonna be comparing the carbon-14 levels in those two samples. And then and that this is with other dating methods as well. But my question would just be like, how do we know that a reference point for things millions of years old is accurate if we can't like have any confirmation of any known things that are millions of years old? Because obviously we weren't there to like witness it, and a lot
What
of
do
it
you
kind of becomes
what do you
this.
mean confirm? right? The same way we can confirm things that we radiocarbon date them forming by just watching them form, right? We can do the same with any of these other methods. There's minerals that form in the modern day that form with uranium in them, the isotope of uranium that decays, right? And so we can take that mineral that we watched form and do the uranium lead decay chain detection on it and work out its age in that method as a reference, right? But like I said, because these are absolute dates, all you're really doing is just making sure your spectrometer is calibrated. But you can do it. You can do it. There's no problem with that. And all of the methods will have ways of working out these references.
So the references are assuming that they have a steady rate of, because we can't confirm anything, any rock is 1,000,000 years old. Like, so whenever we're making the reference points themselves that are supposed to be the like average and
What are you
standards.
talking about? We can confirm the rocks being the strata being certain ages. We can confirm it with multitude methods, multitude different radiometric methods, but also like your fission track and
What is what I was
thermoluminescence
saying.
methods. And we can
I was gonna say,
cross-reference that all those methods converge on the same age.
I got it. Let me finish what I was saying. So this would be assuming that the decay rate is stable over time. And then so basically just to try to steel man what you're saying is just like these decays are stably this we can see a steady decay happening over so many years. We can observe it in a lab. Therefore, if some sample has this much decay, then we can assume or conclude that it's millions of years old. Okay, and I was gonna ask you do like do environmental conditions or could
But it's
conditions on
not
earlier.
just decay
Wait,
rate.
hold on, bro. I didn't even finish asking the question. Like, could environmental conditions or conditions on the early Earth affect decay rates? That was just my question.
Well, it's not just radiometric methods, right? But nonetheless, we're not talking about dating fossils that were anywhere close to the formation of the Earth, right? If we're talking about dinosaurs, that's well after the formation of the Earth, the Hadean, the Archean, all those eons where it would have been hard for life to be around. Right? The
Mm-hmm.
dinosaurs are not in the picture, at least
Well, would
according
things like
to
the Ice
modern
Age, for
evidence.
example, affect them? Like some of the catastrophic events that happened on Earth after the dinosaurs, I'm just wondering if those would affect decay rates.
No, there's been conditions shown like particle nuclear physics types of conditions where the decay rates can be affected. So if the Earth was the sun, then yes, maybe like the conditions akin to the sun can have an effect on decay rates, is what's been shown in one way or another, right? But that's not what modern science, modern biology, modern evolution proposes for that these fossils have never, the strata have never been in conditions akin to this. So the rates are not going to have been affected. But nonetheless, like I said, that would only be the rates of radiometric decay, right? It's also, there's fission track dating, which is a different thing than radiometric dating. And there's thermoluminescent methods, which are also different than radiometric dating. Entirely different decay process of how the measurement is obtained is entirely different than the decay process in radiometric dating. So it would have to be quite the coincidence that all these methods just so all happen to align to the same age for specific
Right.
samples.
So if there was something like an asteroid impact, like happened to wipe out the dinosaurs, I wonder if that could affect the decay because it says like high pressure and temperature can affect like for example beryllium's decay rate. beryllium-7, its decay rate can be like affected by like high pressure and temperatures, like such as like an asteroid impact. So I'm just wondering if the dinosaurs were wiped out by some asteroid or some like ice age or something like that, would they have like still the stable decay rate, or would that alter would that alter it, the measurements? But
Oh,
that's what my question would be, but it's okay. We can
yeah.
move on.
Well, there's a layer of ashes that separates the KPG boundary. Um, I forget what it's an element. I'll look it up in a second. But there's this ash layer, and so that can be dated, right, radiometrically. And then, but what's typically done, if you were to go to like the crater, you can date the ash layer, like I said, anywhere on the Earth, and it all comes back to the same age for when it happened. But you can also date the layers that are above and below that ash layer, right, or where the crater is down in the Yucatan Peninsula down in Mexico, right? You can date the layers that are above and below it, and that will allow you can do that. The layers, not if you go to where the crater isn't, you can date the layers that are above and below the ash layer at that location. And you'll see that all these dates, they all come back, they all emerge, whether you do it radiometrically, thermoluminescently, fission track, or whatever other method you want to use, they all come back to the same thing. That it's a huge problem to say that that's all just a coincidence.
Yeah, I haven't actually looked at the data for this, but I do see that there are some things that can affect decay rates. Yeah, I'm looking at like 4 different methods. Fully ionized atoms, bound
Yeah,
state
the
beta decay,
problem is
chromium, potassium, beryllium, all these types of decays can. I don't know how many different ones they've measured with different conditions. I think that would be something worthwhile to explore. But, yeah,
it's been
that's the only thing
explored.
I wonder.
It's
Yeah.
been explored by both creationists and just regular scientists alike, like institutes like the Discovery Institute, and
Mhm.
the Institute for Creation Research, and what's the other one? Genesis Apologetics. Those kind of places have even done their own kind of research on what conditions will affect these isotopes, right? And so if a valid condition is found, like I said, most of it is like conditions akin to being on the sun. But
Mhm.
if there's any valid ones found, they're accounted for when we're dating if a fossil or a bone or whatever strata is suspected to have been in conditions like that, right? There's evidence that those conditions were present, then we won't use that area, the fossils in that area strata, to date things, because we know that or at least the radiometric date will be off because the conditions for the rate to have been affected were present. So we shouldn't use this area for dating. It's like common sense. You're trying to tell people that science is literally debunking science, but that's not how it works. Other
According to
scientists take
who?
these
Science
things
debunks science all the time.
into account.
Science has always debunked science. What do you mean? We're constantly scientifically progressing. Wouldn't science always be debunking other science? But
No,
anyway, I just want to say,
you're
which dating method do you think is the most
you're
accurate?
trying to say that scientists just wouldn't account for this if these ways that the tests could be altered were found to be that they could be present, or that they could even be possible, that scientists wouldn't account for. We would just we're just going to ignore anything that can affect our test, and that's just not how it works. I'm
You act like
sorry.
everything is like set in stone, like for the conditions of the earth and the catastrophic
No,
events.
that's not at all. I'm not
Okay, well,
saying that
that's
science
the
says
only issue that I
anything is
pose.
set in stone.
Yeah, that's the only thing I was saying, was that, once you're talking about millions of years ago, can you admit that there's some speculation involved?
No, there's no
No
speculation
speculation
about
of anything in millions of years.
radiometric dates. No,
Mm.
there isn't any speculation.
Can you
It's
tell me
a
what the most
simple
accurate dating
process
method is?
with a mass spectrometer. There's
I
nothing
understand.
speculative
I
about
still
it.
need your explanation. No, I know what you're saying. I'm just wondering what you think the most accurate dating method is that's like set in stone. Like which one should we rely on the most?
I would say the most logical thing to do is a combination. You rely on a combination of different types of dating methods and make sure that they all converge on the same age. Otherwise, you only have a certain limited amount of confidence. So the more methods that converge on the same age, especially from different fields, right, the higher your confidence can be in the age that you're getting.
But whenever you do that, they have found that different dating methods can vary in results.
What do you mean? I would have to see, right? There's these methods are very specific, right? The ways you sample, the samples has to be done a very specific way for each method. So do you have like an example of this
Yeah,
occurring?
I have an example, this is which dating method is the most accurate for millions of years old? Because, like, for example, carbon-14 becomes less precise. They say after 60,000 years, but I think
Yeah,
it'd be
yeah.
so that's one example. But what dating method can we use to confirm like millions of years, like with the dinosaur times?
Well, like I said, for us, we use a combination of uranium, lead, argon, potassium, argon, thermoluminescence, and fission track, right? For the what we take the it's also a little bit dependent on the sample, right? It depends on what you have, whether you have a fossil, like a rock, whether you have a strata, which is like the surrounding rock, or if you have an actual bone. So your method is going to kind of, your methods that you choose are going to kind of vary based on what sample type you have, but you're going to take an array of those different methods for each sample, and then that's what we do. And they, so far, we haven't had any discrepancies. They all point to the same either Cretaceous period or the Jurassic or the Triassic for the fossils that we're dating.
Mm-hmm. Okay. Yeah, I'd be willing to look into that more. I haven't looked into dating. The only thing I've looked into is like accuracy of carbon dating, and so that's about it. But yeah, I would love to learn more about the dating methods and how they confirm accuracy. I would just say it would be like I'm just a little skeptical of the millions of years and how and the idea that these like catastrophic events on Earth maybe could affect them. Because we do see in certain conditions, as I named earlier, different dating methods being affected by like environmental conditions. So yeah, I feel like an asteroid impact might potentially mess up some of those decay rates. But we can talk about that another day.
Yeah, I mean, well, again, it's just the asteroid only struck one area. When we're talking about the asteroid that eliminated or caused the extinction of the dinosaurs, the KPG asteroid, right? It hit the Yucatan Peninsula in Mexico. So the crater has been found, so its size has been known. It was iridium, is the ash layer, right? It's found all over the Earth in between layers that always date, no matter what kind of dating method you use, right? They always date to between the Cretaceous and the Paleogene. So it just, it's kind of hard
There's
to
something, whatever wiped out the
argue.
dinosaurs, I'm assuming those same conditions, whatever they may have been, I'm skeptical that they could have possibly affected the uh the decay rate of the isotopes. But
That's
yeah,
why you date above and below, right? So you're dating the rock, the strata that hasn't been affected and the strata above and below it, right? The strata below that hasn't been affected,
Yeah.
right? So
It
if
wasn't like some
because
huge shockwave that
those
wiped out.
conditions didn't last forever, right? They were only a momentary condition on the earth. They didn't last to the modern day, correct?
Mhm. Okay, well, how about this? Send me, if y'all ever do date the soft tissue in your dinosaur samples and y'all get it published or whatever, I would like for you to share that with me. And I would like to look at like the dates, the radiometric dating on the soft tissue in the dinosaur bones. If y'all ever do that. Because I haven't heard of anybody radio dating these soft tissues. I think that would be worthwhile.
Yeah, yeah. Like I said, so far we have like a few preliminary positives, so it's going to be quite a long time before any kind of like official publishing would happen. Especially like, I mean, the radiometric dates and that procedure will probably be included somewhere in there, but that's not going to be the focus. If it, if it pans out, the focus is going to be finding either the soft tissue or the genetic material ultimately is what we want to find, right? So that's going to be the focus, but there obviously we'll have the procedure for how the things were dated, but this is probably, I would say, at least a year at minimum in the work before any of that is going to get released.
Have you found consistency between different methods? Like, well, I'd like to
Yes.
see your results. That'd be really cool. Well, if you don't put them in the paper, maybe just like as a scientist friend to another scientist friend, you could share them with me. But yeah,
Yeah,
I'm
yeah. The
definitely
ages
dating more.
the ages will be in the paper and the procedure, there'll be a reference for the procedure, the type of dating methods that were used. Those will all be in there. I'm just saying they're not going to be that's not the focus of the study, but they're part of the process and they'll be included. And yeah, when I can release them, I'd be happy to.
Mm-hmm. Yeah, and so like just looking here, they're just looking at some of the uncertainties, like with the uranium dating, like they assume the known initial conditions. That's one that's unprovable since there are no observers. The decay constants, uncertainty in the decay constants in uranium isotopes affect the age of calculations. Non-radiogenic lead, I don't even know what that means, but the presence of common or initial lead of unknown composition complicates age determination.
Again,
Contamination, radiation damage. Okay, so alpha particle emission apparently will cause damage to
but again,
affect.
you're going to science, right? Some of that's not even true. I don't know where it's coming from, but you're going to science to
Okay,
try
look,
to
there's
debunk science. Like
science debunks science all the
we
time.
already know that this stuff
We don't know
that you're
anything. We can
that you're
assume.
pointing out, right? We can all do the same thing, right? When we're doing these dating methods, we can all go to Google and look at these papers of these potential risks that are involved. We get together at conferences and we talk amongst each other, and these things get brought up, right? So we're aware, right? You're going to science, published science, to try to debunk other published science that's
Good, they
already
do that all the
accounted
time. Scientists do that all the time.
it's already accounted for the original published science. Like it's just not how
Okay,
it
I
works.
want you to know that scientific papers are always debunking other scientific papers. They're constantly trying to like
No,
there's
they're
many papers
not.
that are posted as responses to certain research and criticizing them. Like upgrading, basically saying like this was a better method, or pointing out issues. They're always doing that. Science is always doing that. Like why would we assume that it wouldn't do that with aging and dating these samples? They should. They should be. We should be critical of other science. That's what science should be. Like a pursuit of knowledge. And in that pursuit of knowledge, you're going to be critical of things that are considered like fundamental truth. That's very fine. That's fine. It's okay to ask questions. I think that's okay, but maybe some people think that things shouldn't be questioned as much or whatever. But I think that we should continue to question the quote unquote general, consensus in science,
Yeah, you're just misrepresenting what I'm saying.
Well, I'm
Right?
not trying
Not
to.
every scientific paper is debunking another
Not
scientific paper,
everyone, you're right.
right? There's plenty of literature reviews, which are just papers that just summarize. They take a lot of the data, experiments, observations in the field, and they just summarize it all in an easily digestible way for other scientists. So they only have to read through one paper rather than 400 papers, right? And then there's
I was talking about
also
literature reviews.
dissertations. Yeah, literature, that's what I said,
Why
literature
are we talking about
reviews.
literature reviews?
There's also dissertations and PhD theses or undergrad theses, which are presenting novel research. It doesn't have to be debunking any old research. It just has to be a novel topic, right? So not
Okay.
every paper in science is cutthroat debunking another science
Not
paper.
everyone, but yeah. So like, for example, like with the origin of life, when I talked to Grayson Hawk about, his theories on the origin of life, he proposed the amyloid world hypothesis. And the science that's basically like a protein first mechanism. And they in these papers, they were mentioning like the issues with the RNA world hypothesis. And so like they're like, oh well, even though like most people do believe in RNA world, they were posing a bunch of issues with it, which is fine. So they're kind of in a way going against the mainstream narrative, but proposing another solution at the
No,
same
they're
time. So,
not, because those
oh, they're not.
the
That's weird. Did you read
potential
the paper?
risks, the potential problems with the RNA world are known. That we know about them, right? That's why I say I don't subscribe strictly to the strict RNA world hypothesis, right? That's not what most modern day origin of life researchers would subscribe to. But a lot of them subscribe to a modified version of that accounts for the problems that have been identified, or the potential problems.
Yeah, I was just actually reading this paper because on the formamide based RNA hypothesis, basically where they were using like formamide chemistry to make RNA, and then there was a paper that I'm trying to find the name of it, but they were basically like it was a paper about why this is like unreasonable and like the issues with it. And so like that's common. Like you're gonna have scientists like questioning things and questioning the accuracy of things, and that's just
Yeah,
fine and dandy. And that's the only
I
thing I was
wasn't
doing with
saying.
the dating methods. I was just questioning like if these other environmental conditions could potentially like, impact them, and have we, been able to. I know you're gonna say we probably like studied it, but I'm just I would like to read more about how accurate they are and what assumptions they are making and some of the other questions and problems I just posed, which is fine.
Yeah, and you should, but I wasn't saying that in any way, that science doesn't question other science and test things and verify things. Of course it does, but you're trying to sell the idea that the people doing these radiometric dates for fossils and bones and strata, geologists, paleontologists, etc., right? They are getting these ages and not accounting for these other factors that have been published by this other science that you're referencing, these certain particle physics things and nuclear physics and other kind of geology and environmental studies, right? You're trying to portray the idea that we just haven't, when we do these ages, we just haven't taken any of that into account, and we just make these large assumptions, and it matches what everyone else is saying, so we go with it. And that's just not how it works in any way, shape, or form.
Right. I mean, I just think that they would even acknowledge that there are assumptions being made in these dating methods. Like, even whenever you say they're accounting for environmental conditions, like, how would they know the environmental conditions? Like, how would they precisely know exactly what happened if they weren't there? They have to, at some point, be making some assumptions, even if they're going to say, oh, we see this in the rock, and therefore we're going to assume this was the environmental conditions. So, either way, they're going to be making assumptions somewhere, and they don't deny that.
No, because paleogeologists have been studying what these potential conditions could have been for hundreds of years now, and we reference what they've concluded at the most up-to-date times for conditions, if for these strata or for the fossils, for the bones, or whatever you're finding, right? People have been studying the conditions that those things are found in for hundreds of years now, and they've put forth great hypotheses with evidence for what the conditions could have been at that time. And so we use those sometimes to calibrate the spectrometer, sometimes for other purposes, but we take
Mhm.
into account all of those things when we do our study.
Yeah, so here's one example of an assumption they'd have to make. They'd have to assume that, like, the original composition of the sample, like, they'd have to assume what the, like, original composition of some, like, you know, carbon-14 may be or uranium. They'd have to assume, like, the original sample has this amount. So, like,
No,
they acknowledge that there are
what do you
assumptions.
mean? The radiometric dating tells you how much and how old the sample is, like
But how
through
much
the
original
spectrometer and
carbon-14
the
do they have?
math. Yes,
They're
it
assuming that
tells
this
you.
particular sample
No, you
started with
don't
this.
have to assume. It's simple first order chemical kinetics that you just apply to isotopes, right? It's simple math. You can work out from the age that you get, you can work out the original concentration, and then you can take into account all these other factors when you look at dating that's been done and study that's been done on the strata, right? So if you're dating a sample, a fossil, a bone, whatever it may be, right, and you think that your age might be a little uncertain, you can't quite work out the original isotopic amount that was in the sample, right? You can then go to these other methods, dating the strata and whatnot, to work out the conditions and work that
But
stuff
along
out.
the way, there are assumptions. You can't, I mean, it's hard to see that you're denying that. I mean, it's just like, that's crazy. So they are acknowledging assumptions whenever they're doing these radiometric datings on many things. But yeah, I mean, we can try to find, you know, the best, most accurate way to date it and the best results. But along the way, whether you're going to be assuming the age of another known sample or assuming the age of the rock layer, or like, you know, these reference points, or assuming the amount of uranium from the original sample, things like this, there are some assumptions that are being made.
Well,
Even
being someone who does this like all the time lately, I don't make any assumptions when I'm doing it. It's strictly an instrument process, right? There's some chemistry to it. You know, you got to have a little chemical knowledge and savvy to prepare the sample. And then it's simply putting it through a mass spectrometer. So you have to understand
Mhm.
how a mass spectrometer works, right? And
No, I do.
first order isotopic kinetics. It's literally like there's nothing to assume. It's just a procedural process that results in an answer and an uncertainty.
Yeah, so, like, for the, like, uranium lead dating, like, how would you definitively say that this sample originally had this much uranium lead in it? Like, of, like, the
What do you mean?
uranium
Number one,
238
you
isotope?
can work out the age without knowing the original concentration. But number two, like I said, it's just first order chemical
I
kinetics.
kn
You take
I
a
understand
measure
you're
of
looking
the,
at the decay of the uranium over time, but how would you know the amount of undecayed uranium in the original sample? Wouldn't you be assuming?
what do you mean undecayed uranium? There's different isotopes of uranium, and that's why
Mhm.
you do something called an isochron at some point on your sample, which is comparing the different rates of decay, the rates of decay for the different uranium decay chains, right? And so that's a way of kind of working out the original concentration of the isotopes in a multifaceted way. You're both doing it through the different uranium isotopes, but you can also do it using just the first order kinetics, right? You measure the amount of uranium that's in the present sample, right? And it's just first order kinetics. You can work back the uranium and the lead. You have to measure the decay chain that occurs. And from that, you can work back using just a simple equation. You're literally just solving for a variable. It's just algebra to work out the initial concentration. And then you can use isochron to do it using isotopes to get the original concentration to make sure the two match. Both your math matches and your measured amount matches with the isochrons.
I see. So let me just read one sentence, and I just want you to tell me if this is accurate or not in terms of uranium dating. So initial composition assumptions, accurate dating requires estimating the original amount of parent and daughter isotopes, which can be challenging. Is that true?
No, it's literally what you're measuring. You can literally measure the amount of both parent. You literally are measuring the amount of both parent and daughter isotopes. That's the whole thing.
How
I'm
would we
talking
not?
about the
Yeah.
decay chain. The decay chain that you measure, right? So it's not an assumption, it's literally the point of the whole thing that you're doing is to measure those things.
The amount of parent and daughter isotopes, I get it, but how do we know the original sample didn't have more daughter isotopes than what we'd assume?
Oh, like I said, there's multiple ways. The first one is there's a mathematical way to get the answer. Then there's the isotopic way using the isochrons, like I said before. So that's an actual experimental way. And then there's you can also look to environmental factors if you need to adjust the original concentration because something in the environment was present that would be affecting it. So people do go to those lengths to even do that, to study the environmental strata and to make sure there's nothing there that indicates you would need to account for something affecting the uranium decay rate or the lead decay rate, the helium emission rate, whatever method that you're using, the thermoluminescence emission rate, doesn't matter, right? All that can be accounted for.
So whenever we go look in the strata, do you have an example of something that we could like see to like make an assumption on the environment then?
What do you mean? Something that would indicate that the original concentration might be different than expected?
Yeah.
Yeah, there's certain things, certain geological events that occur. There can be like algae blooms that kill off a whole bunch of organisms all at once in the ocean. And so that can affect the carbon rate, the lead rate, the uranium, the argon, the potassium, can affect those rates. And so when we see these kind of events happening, that's like a biologic, geologic event. But there's also just like where minerals will get into the water in huge amounts by some event, some geologic event occurring, an earthquake, a mudslide, whatever in some region. The water just gets all this mineral stuff injected into it all at once. Well, that's going to throw off uranium, lead, potassium, calcium, all those kind of rates. So when we see these things happening, when we study the geology of these areas, we make sure to account for them when we do our biological dating studies.
So whenever they do like look at the mineral rates, for example, in different strata, like how would they then say, okay, well, this mineral, like I guess they would be assuming that this event caused it or whatever. Either way, the mineral's there. How would we know like that this mineral would cause this much of a change in the decay rate?
Well, it's not that it causes a change in the decay rate. Like the cause, when we're talking about radioisotopes, the types of changes, um, the conditions are, like I said, akin to the sun, right? So that's not it's only that's only going to happen like right at the site of where an asteroid occurred or something of the sort, a volcanic eruption, something drastic, right? And so we know just where we see those events, not to date not to use those for certain methods, right? Because they may have thrown off the rate. But I'm more talking about where the background environment amount of an isotope or uh let's just stick with isotopes. An isotope is there's higher than like one area has a higher percentage of that isotope or of that element compared to the rest of the area or the a continent or the world, you could say, if you want to take it that far, right? That can cause a change, that can cause a error in your measurement when you're looking for the age. It doesn't cause a change in the decay rate. It causes just environmental contamination. This isotope is just higher in the background of it, is just higher in the environment than usual, right? And that stuff has to be accounted for, and it is accounted for.
Okay. Yeah. So I'll send you this book. It's called Geochronology in Canada, and it goes over all the limitations and assumptions that would be made in radiometric dating. Maybe you could check it out, and
Yeah,
I'll definitely as well more thoroughly, and we can maybe talk about it in the future. But I do think that scientists do acknowledge, limitations and assumptions that are made in these dating methods. I didn't think that was disputed until until now, which is what you're saying is that it's undisputed and there are
disputed.
no assumptions.
I'm not disputing that, right? We do acknowledge limitations and assumptions. What I'm disputing is the implication that when these radiometric dates have been taken, be it by myself or by some other group or some other researcher, and published, that we're not accounting for and acknowledging these limitations or assumptions if they're present, because we absolutely do, and there's multiple examples of that.
Okay, well then we agree. Then we agree that as long as we agree that there are some form of limitations and assumptions. That's the only thing that I was positing.
Well, no, because I don't agree, right? I agree to an extent, but not to the extent that like the dates I'm getting in my lab are wrong, or the dates that other people that match exactly other people or myself, right? Cross-referencing, that those are wrong. Like that there's some kind of limitation or error that we're not acknowledging, or something that we're just assuming, yet somehow coincidentally still all getting the same
Well,
answer for multiple
if
methods.
there was some catastrophic event, then it would affect all of the. If some catastrophic event did cause like the decay rate to increase, then that would affect them all the same, and you would get the same results still. But anyway, like and if you're using the same reference points, then that could also account for why you'd be getting the same, you know, same dates. But yeah, I feel like we can talk about that more in the future. I'm gonna learn more about it. To be
There's
honest,
been, in the
I
present day,
was
there's been a lot of things that have been radiometric dated now, things that aren't even from the Earth. Asteroids, meteoroids have been dated as well.
how can you date like a rock, for example? Because like I'm
Because
honestly
they
I haven't
contain
like I said,
isotopes.
yeah. Let me finish what I was saying. I was just saying like that. Um, I haven't taken any like geology courses or anything. I don't. I have never really studied radiometric dating or any sort of dating methods to an extent, obviously, that you have, but I can just share what I've learned in that, some of the assumptions that are made and the criticisms of it. But so that's kind of why I have these questions too. But in terms of rock, I would think that I'm just curious how they would date a rock because it seems like the isotope levels would reset like in the formation of the atoms or something. And so like when they were first created, these elements like in a star. But I'm wondering like in a rock, like how would we reset that isotope level to like date it? Just
Because
my own curiosity.
the elements that make up the rocks on
Uh-huh.
Earth haven't come they were in the accretion disk that the Earth and the Sun formed from. So the molecules in the atoms that make up the molecules that make up the rocks, they never went back into a star or came from another star after the Earth formed. That right? They came from the star first. Then the Earth formed and utilized their presence in its formation. Right. So the
Yeah.
whole
So whenever
premise
we
of
date a
that,
rock in space, are we dating it from the time that the rock formed in space? Like,
yeah, it would be. I guess, like I said, it's every sample requires different accountizations. I guess you could say different things you have to account for, right? But yes, you're basically you'd be dating for some isotopes you're dating from the last cooling point, right? It's from when the sample reached a specific temperature, it came below a specific temperature. That would be when the clock starts. And some
okay.
isotopes are just when they originally formed, like helium, hydrogen, things like that. The lighter kind of isotope. Um, you can date those back in the space rock or the asteroid, the meteor, or whatever you're dating.
Okay. So the isotopes reset whenever the rock is melted. Is that what you're saying?
Some isotopes can be reset when if a rock or just a sample in general, it doesn't matter what rock it is, what kind of rock it is, or even if it's not a rock, there's certain isotopes that if they get above a certain temperature, then they start to have changes in the decay rate. And so those have to be either accounted for, or most times you just don't date those samples that have been through conditions like that.
Yeah, I don't know how we would know whether an asteroid, like when the last time it was heated up, but it's interesting.
Well, right. So I think the conclusion was come to for the ages of some of these asteroids by dating with multiple methods, multiple different asteroids, right? Like you get a date, you get the methods, they all converge on one let's say a meteoroid, because you have it down on Earth. So you get one of these meteoroids, you do your different methods, and they all converge on a date. Then say NASA does they collect a sample, or the European Space Agency collects a sample on some asteroid from somewhere, out in space in some known region where there's multiple asteroids, right? They collect multiple samples, right? And then you can work out the ages, right? If they bring them back, then they can work out the ages, or even, I guess they probably have methods nowadays, spectroscopic methods that could work out the ages of some of these things. But for geologic purposes, I would go with they bring a sample back of an asteroid, even ones that they like intersect. I know they've intersected a comet before. I'm pretty sure they've intersected an asteroid, and so they can get samples to do analysis, right? And so you get multiple. The point is, you get multiple samples from multiple sources, and you compare them with multiple methods, and you account for multiple different things, and whatever falls out as the result of that is the answer.
Doesn't sound concise at all. But it's cool. Like, I just don't get it,
Well,
like,
it's
how they're
because
knowing
I don't know
approximately.
the exact, like, what exactly they've dated to do all these references, right? Like, you'd have to look through the literature to find that, right? So I'm just trying to give you the overall picture.
Mhm. Okay. Yeah, well, if we come to the conclusion that there are some limitations and some assumptions that are made, that's the only thing I was saying, but I do hear your point in that we can do multiple methods and we can get the same age or whatever. So, yeah, I hear you on that point.
All right. Well,
Do you have
I do have to take
time
off
limitations? What'd
shortly,
you say?
so I have to take off shortly, so we can
Okay.
I mean, if there's something you know, that you really wanted to talk about, we can. But we should start wrapping up shortly.
Okay, I'm sorry if it wasn't a good time, like.
Oh no,
Mhm.
it was fine.
Okay, cool. Well, I was just gonna ask you about, like, in terms of original life research, if there was any, papers showing that we could make, like, the self-replicating RNA just from purely nucleotides, like, without adding template sequences, or, like, if this is a feasible theory that you say that you hold to.
Yes, so what you're talking about, right? You're talking about the intersection of where systems chemistry intersects with autocatalysis, right? So that would be where the area of research where you're going to find the published studies and experiments and whatnot
Mhm.
about that, right? I
Yeah.
know that there's certainly some. I'd have to Google them and look through it a bit to refresh, but absolutely the autocatalytic cycle or the self-replicating of RNA it's no mystery. It's systems chemistry. The
Yeah.
complicated details have been worked out and they're
Yeah, it's just,
published.
like, demonstrating it from the bottom up, you know? Because, like, in the self-replicating RNA, that the papers that you posted in here before, like, they've used, like, for example, RNA polymerase or, like, a template sequence they've inserted, like, stuff like that, but, I think that they should be able
They
to
didn't.
demonstrate. Huh?
They didn't, right? That's how we'd have
Oh,
to
they
get
didn't?
specific. What paper specifically?
In the self-replicate, like, the ribozyme paper, like, for example, they use, like, a RNA polymerase to have this, like, autocatalysis instead of, like, starting from, like, prebiotic conditions, and they acknowledge that.
Okay, so run me through this, right? These are A-Biogenesis, origin of life researchers
Mhm.
researching how RNA would have become autocatalytic or its autocatalytic cycle or aka become self-replicating, right? And so they're looking for the mechanism of how that would have formed. So they use something that shortcuts that entirely by using a ribozyme or a polymerase or a template. And that makes sense. That doesn't make any sense, Taylor.
Yeah, so, let me give you the name of the paper.
It's not a logical thing to do what you're claiming. No origin of life researcher would do that.
Oh, really?
Yes.
Okay.
If they're
So
looking, no, hang on, because it's not
okay.
just the in general, yes. It's if they're looking to discover the autocatalytic cycle mechanism for RNA or DNA or any kind of genetic material, right? They're not going to do that because that defeats the whole purpose of
Mhm.
looking for the its natural autocatalytic cycle. So this just
Okay.
doesn't make any sense.
Yeah, so the papers about the self-replicating ribozymes, like they basically use like an RNA polymerase and like modify the sequence in the class 1 RNA polymerase to get a population of 100 trillion molecules. From this population, they accepted ribozymes. They could transcribe 2 different RNA molecules with a complex 3-dimensional structure. And then they identified the ribozymes with the desired properties. They repeated
Yeah,
the process, mutating the
because
newly
Taylor,
identified
hang
ribozymes
on.
to produce new populations of molecules. And after 24 rounds, they had successfully evolved a ribozyme they called the 24-3 ribozyme that can copy RNA molecules with complex 3-dimensional structures,
Yeah.
and in turn
Okay,
make
hang
new
on, hang
molecules.
on.
But the point was that they were
Taylor,
starting with an RNA polymerase.
Taylor, it's because that study that you're talking about, they are not describing the mechanism for the step of RNA itself, just an RNA strand becoming self-replicating, right? They're not talking about that step. They're talking about how after RNA became self-replicating, how it transformed into something akin to a ribozyme to have certain functions. So they're testing in that study, in that part at least that you're talking about, they're not testing how an RNA strand started self-replicating itself, even though that could be the result of the ribozyme, right? That the ribozyme can help an RNA strand self-replicate, right? But they're not taught, we're talking about abiogenesis here. So they're not talking about that because they would need the RNA to come about without a ribozyme, because the ribozyme is already way further in the process that's hypothesized.
Well, have they done
So
that?
they're talking about after the RNA has already gone through its self-replication mechanism, its autocatalytic cycle, and
Uh-huh.
you have these RNA strands now. How do they interact with each other based off the different sequences? Because every time they self-replicate, they have slightly different sequences, right? So in these autocatalytic cycles, when they have the different sequence RNA strands that result from this, how do they interact with each other? How can it be that they can turn into something like a ribozyme that the modern day cell has? All right, so that's what they're studying. And so when you're studying something like that, you can just, you don't need to have the RNA come about naturally, because that's not the step you're studying. So you can use a polymerase or a template in something like PCR to amplify the RNA strands in that case and just synthesize them yourself, however you want to do it. They don't need to come about naturally, because that's not the part of the process that you're studying. You're studying about how, once they've come about, how they interact with each other and form more further complex structures.
But if this is going to be invoked for abiogenesis, like I saw that you shared some papers about it, but like I think that if they're not starting from prebiotic conditions that would be relevant to abiogenesis, that shouldn't be invoked.
Well, I shared that paper not for that specific claim, probably because I shared that quite a while ago, and I'm sure we weren't uh, talking about this specific, like how RNA strands became autocatalytic, because I wouldn't
Mhm.
have shared that paper then, right? There's
Okay.
plenty of work out there that's been done on the autocatalytic, the abiogenesis, or the prebiotic autocatalytic cycles for RNA that are in the field of systems chemistry.
Okay. So I'll just like tell you like some of my issues with original life research, and then maybe you can respond to them if you want. And then so basically, all right. So basically, like the different conditions, like to form the proteins and to form the RNAs. Like, how can we have like a unifying like place or condition that this could happen in, like, or like at least a unified model of like, okay, this happened in this place, this happened in this place, and somehow they migrated and came together, so on and so forth. And then once they migrated and came together, like another problem I have is some of the incompatibilities, like in the conditions um and in the actual molecules themselves interacting with each other. So like first of all, like um things like the amino acids, can react with like sugars in something called a Maillard reaction. I think that would pose a problem to like the proteins being
when you're
synthesized at the
cooking
same time as the
steak,
RNA. And then also like the translation between the RNA to the proteins. How did we ever get the translation system to
all
come
right, hold
out?
on, Taylor. You're right. All of these things are like extremely complex, and they're all different points of the process, right? So the thing about the Maillard reaction is like what happens when you're cooking, right? These molecules obviously wouldn't form, they don't form proteins, at least biomolecules, proteins, nucleic acids, carbohydrates, and even some phospholipids. They denature, they fall apart under those high temperatures. So obviously they didn't form in conditions where you had those high temperatures
Have you
like
looked at the
that.
temperatures that are required for the RNA synthesis for the nucleotide synthesis that would be required for RNA? They literally invoked an asteroid impact.
No, they
For
didn't.
for
There's
the
been
experiment with
plenty
John
of
Sutherland, they shot it
groups.
with a laser. These would have high temperatures, but anyway. Look
There's
at the
been
temperature
plenty
requirements
of
for any paper
groups.
that you think they can synthesize RNA under.
I have, and
Okay, what are the
there's
temperatures
been
then?
plenty of groups. I brought one up, David Deemers, who have done this even just out in nature, right? They're not, the conditions are, they're not, they're not anything abnormal. You can do the synthesis. I'm sure there has been people who have done the synthesis under either using lasers to kind of emanate temperature or emanate energy, or they just use the temperature to emanate the energy. So they do it at high temperature, right? Because they're trying to test, like, maybe this happened under these volcanic conditions. This one part, one step, or one portion of the process may have happened. So they're at that temperature. So some people may be hypothesizing or trying to work out and demonstrate how it can be done at high temperature. But it has certainly been shown just under lab and natural
Speaking of
conditions using
high
just
temperatures,
catalyst.
the paper by John Sutherland to produce the nucleotides, the temperatures reached 3,500 to 7,000 Kelvin. They had to use a laser. They invoked an asteroid impact for that to happen, by the way.
Okay, well, that's not the only paper where nucleotides were produced. And I'm guessing that was part of some kind of cycle that he was doing for a specific purpose, and not just to overall claim that nucleotides can form under whatever conditions, because people have done it with catalysis instead. Systems chemists have done it, and biochemists and stuff have done it using catalysis, using montmorillonite clay. We talked about this the last time I was here, right?
Yeah.
And that you don't, using mineral catalysis. So it's not just limited to this one clay, but you don't need to use these crazy temperatures.
So, like, I'm
You
talking
can
about to get
get,
activated nucleotides, and with the mortar-millionite clay, like, they used already, like, activated nucleotides. So this is, like, creating the nucleotides themselves, if you've
my
looked into that
God.
before.
Yes, obviously they used activated nucleotides, because all that means is that they used nucleotides that have the ability to react, right? Because that's what would be needed in nature for this process to occur. They can't be deactivated nucleotides, because if they can't react with each other, they can't form RNA strands,
Uh-huh. So the
right?
high
So
temperatures I was talking
they
about was
all,
creating the activated nucleotides.
no, you don't have to use high temperature. That's one way of
Do you have an
doing it.
example? You
The
said
clay,
David
the
Morton,
Merlinite clay, is used as a catalyst, but there's
but you're not
other
making the nucleotides
mineral.
in the mortar-millionite
Yes, you
clay.
are. Yes,
No,
you
they
are.
use activated nucleotides. I literally just told you that.
Oh my
You're not
God.
creating activated nucleotides. All they're doing is bonding them together.
Oh my God. that word doesn't mean anything, Taylor. Activated versus deactivated. They're using just nucleotides in their natural state with the ability to react, how they would in nature and how they do in chemistry. They're not using deactivated nucleotides, which would not be able to react with each other and thus could
Yeah, that's
not
why so
form
they're framing it for the
RNA
oh,
strands,
but
right?
no,
So they're
okay, so I'm talking
using
about getting created activated
they're
nucleotide.
using RNA in its natural state, in its with its ability to react as
We're
it's
talking about on
found
prebiotic Earth.
in
You have to make
organisms
those nucleotides. You
and
need to
in
make it with the
nature.
phosphate bonds to react.
And then they
And so
use minerals, which in some cases can be found in the montmorillonite clay. They use the phosphate minerals in there to catalyze the reaction that polymerizes those nucleotides into RNA strands
exactly.
without
So can you tell me how
without
we would get these
temperature.
nucleotides? Like, can you tell me how we'd get these nucleotides without high temperatures?
They come from prior reactions, the autocatalytic cycle that forms nucleotides. And
But wouldn't you have to have the same conditions that you're creating the nucleotides in order to bind them?
no,
Oh, so
no.
did they get
The
created in
the
another location and then
forming
they went to another location to bind?
the forming of ribonucleic acid or just the nucleotides itself, each individual monomer can occur in separate conditions from the polymerization of those monomers together. That's going to occur under different conditions
Do you have a
because
hypothetical of how that could happen?
no, it's just how it's done,
It's
even
just
by
how
chemists today.
how could that have happened on early Earth? These are forming in one place, but they're needed over here in a different place in different conditions. This is just for RNA creation.
Ah, well, yeah. Right. So, oh, it's just these things happen under certain conditions, right? And then out in nature, environments mix with each other. Salt water mixes with fresh water. Tide pools get absorbed back into the ocean. Uh, Certain the desert eventually turns into grassland. It could transition into rainforest. Right, the environments cross with each other,
There would be
and
no grass or rainforest on early Earth.
I know, I'm just giving you an example, right?
Okay.
These different areas, yes, they're not going to have living organisms on them yet, obviously, but different areas of the earth will still have different inorganic or non-living conditions. So different water versus like soil conditions where it's just, minerals, crushed up rocks and stuff, forming continents and whatnot. There'll still be like beaches, there'll still be intersection, rivers intersect with oceans and stuff like that. These environments will cross with each other, but usually, right, going from just the RNA monomers, right, because up to that point you're really doing monomer reactions, right? You're not doing polymerization reactions. So up to that point, you kind of, the conditions will vary, but they're not going to be like extraordinarily different typically.
I think they
But
would be.
when you're going to polymerize something, getting a polymer together is a much harder chemical process because you're linking a bunch of monomers
Yeah.
into, which are already quite big when you're talking about biomolecules. These amino acids, nucleic acids, lipids and stuff, they're already pretty big molecules, and now you're linking them together into even bigger molecules. So yes, that is going to require different conditions, but we're talking about just the formation of these basic biomolecules, right? The further structures come further
So
down the
let me
line.
respond at this point. You're kind of going on. So let me just say this. So for the nucleotide formations themselves, like not just the binding of them on the mortramillionite clay, they had conditions around 55 to 85 degrees Celsius in which they were able to create the nucleotides. And I did want to mention that Maillard reactions can happen in temperatures of 55 degrees Celsius. It would be slower, but the reaction I mentioned earlier would still apply. I just want to let you know that.
Okay, but right, the Maillard reaction requires it can happen, sure, at some slow rate under kind of low, what we would consider low temperatures, but it usually requires higher temperatures. But nonetheless,
Well, we would have
it's
very high
not a
temperatures for like the
factor.
protein creations too, by the way.
It's not really a factor because we're talking. We also have to like think about the eon, the archeon eon, right? It's before there's living things, so the environment is quite different. Like the atmosphere was different, the ocean was different, fresh water was different, the land was different, right? And so all those things have to be taken into account. Like the
Okay.
chemistry was
No, I
more
got you.
active.
I'm just saying, like these types of cross reactions in different conditions for the different prebiotic molecules to form seem very contradictory if you look at the papers and research on them. So they would at the very least
I
have to
don't
involve
think
multiple.
so. It
Okay, well
seems to
give me one
be
example of the
there
paper and I'll
there
give you multiple other examples of another process needing different conditions.
well, what do you mean? Nobody's denying that certain steps need they all have their own little conditions that are gonna favor certain aspects.
But eventually they'd have
Right?
to
Like
come
you're
together and
trying
be
to
synthesized
boil
in the
it
same
down.
location.
You're trying to boil
But
it
but at
down
some point they would have to
into
come and be
something.
active in. Go ahead.
Hang on, because you're trying to boil it down way too simplicity into too much simplicity. It's not just some linear process, right? It's kind of like a environment interacting with chemistry, right? Especially this part we're talking about with these biomolecules. That's like the complex intersection of the early Earth environment with the early Earth chemistry that was occurring.
Right. Yeah.
So you
So what
have
I was
these
gonna
molecules can be forming in multiple different environments and getting different environments have a different part of the process or have gotten to a different part of the process, and then they can intersect with each other. So it's not just this linear thing, it's the whole entirety of the earth. We're trying to figure out what happened in reality, so we have to account for all of reality to make sure we're getting the most accurate picture that we can.
Right. Gotcha.
And so these
So
RNA,
basically,
you're pointing
oh
to
my
these
gosh, you have
papers.
to at some point, dude. I have to be able to respond. I'm
The point is you're pointing to these one paper about RNA at high temperature and
no
trying
multiple
to
papers. I'm referencing about 12 papers.
imply that
I tried
that
to find
is
all the
the
best papers in
only
your
way, and it's not.
dude. Can I respond, please. I'm trying to find
I
all the
wasn't
best papers.
done.
Dude. I'm trying to find all the best papers in your position and for your favor. Like, what is the best experimental evidence for abiogenesis? Okay. Like for the creation of proteins, for the creation of RNA, nucleotides, amino acids, things like this. Find the best ones that can actually explain how they would be created in prebiotic condition. Then you compile them, and I have them compiled on this Word doc that I'm looking at. And then you look at the conditions of each reaction, and you see that they required different conditions. And that's one of the things. So then you're saying, okay, well, they could form in different locations, but then
What do
can
you
you
mean they
see how that
require?
they would they would have to come together at some point in order to, have a continuous supply of nucleotides and amino acids for proteins and RNA and stuff like that. So they'd have to at some point be able to converge and work in a cohesive fashion. And our cell has a great way to, well, first of all, they have enzymes, so you don't need these different conditions. We have the enzymes to overcome like the activation energy needed for these bonds. So that's one amazing thing. And then they also have compartmentalization where different reactions and processes occur. And yeah, but in the prebiotic earth, you're going to have to invoke all these different crazy environmental conditions for each individual part, and then getting them to come together cohesively would have to happen at some point.
All right. Well, I do have to take off in a minute here, but you're just oversimplifying is the problem with
No, I'm
that,
compiling it.
right? You have
Compiling the data
no, you're not
and look at
compiling
them
any
see if
data. You
they're
haven't
compatible.
given any specifics. Who did what under what
Yeah,
conditions?
for
What
example,
was
the
the
RNA
result?
polymerase with multimillionite clay, that was one of the conditions that I was just talking about.
Yes, yes, that's been done by multiple people. You even pointed to a different type of formation mechanism where RNA was forming under high temperature.
They would need, that's what they're seeing that they need for like the actual RNA formation themselves, like with John Sutherland's paper in formide synthesis of nucleotides, which make up RNA. So just any of the papers, really.
Did he use the temperature or did he use catalysis?
No, he used both, but yeah, they require different temperatures for these catalytic rates to occur.
No, temperature will have an effect on the rate,
Mm-hmm.
sure, but it's not required.
Well,
It
they did
doesn't
test them in,
necessarily mean like 20 degrees Celsius is going to have a significantly different rate than 30 degrees, right? Just every reaction has a rate.
okay. And actually, like he was actually trying to make some prebiotic molecules for nucleotides as well, and he couldn't do them in a one-pot reaction. They required a different reaction. Like they would require different conditions. So
Sounds
it's
like he was testing one mechanism
he was
with
trying to make a one
formamide.
of amino acids in RNA. That was his goal. And he was using formamide, and he was saying this could be like some precursor. But then for the portions where they did the RNA synthesis or synthesis of nucleotides, they were different conditions. They didn't do them in a one-pot reaction. They required different conditions. I don't think anybody would deny that the protein amino acid synthesis requires different conditions than nucleotide and RNA synthesis.
Yeah,
So
proteins have been made. I know for sure there's been wet dry cycling done to do the protein synthesis abiotically or prebiotically. Um, Nucleic acids, I'm only aware of like temperature variations and catalysis because these molecules they arise just by the thermodynamic properties of systems chemistry, right? So
yeah,
the temperature doesn't have to be involved in that though, right? You can have mineral catalysis, which is what the Montmorillonite clay, but you can catalyze the reaction. It could have happened prebiotically other ways than just this clay. The mineral, it's a chemical that's contained in that clay that catalyzes the reaction. And that chemical can be found in other
yeah.
substances in geology.
That's just for
Right.
RNA polymerization. No, that's not for nucleotide synthesis, which make up with RNAs for the audience. I just want to mention that. So, yeah.
Well, polymerized RNA nucleotides is RNA.
Yeah, no,
Polymer,
so I'm saying the nucleotides that make up the RNA that you're referencing for your micromillionite clay were made in different conditions than binding them together for
yeah,
the clay experiment was just for binding the nucleotides, not for synthesis of them.
yeah, it's for polymerizing, for
Yeah, for
catalyzing
polymerization.
the
You're exactly right.
polymerization reaction.
Mm-hmm.
Yeah, yes, but the forming the RNA, that's all systems chemistry and autocatalysis. I
So,
don't
for
know
example,
what John
like I was
Sutherland's involvement in that is.
no, I
I
just
don't
referenced him
really
because he made
know.
like huge like breakthroughs recently in quote unquote in original life research. So, um, I'm just trying to use like the best papers and the most like recognized ones that have the most progression in any original life research. So that's why I was mentioning his, but any one of them would be great. And so I like made this chart too about like the synthesis requirements of like these different experiments that were able to like make RNA synthesis and protein synthesis. And like in the protein synthesis, they needed temperatures greater than 100 degrees. And then so for the RNA synthesis, they needed much lower temperatures, like at the one in the micromillionite clay that you had referenced. giving like another like
Yeah, I think
incompatibility.
because you have the wrong idea. It's not that genetic material like RNA had to form in one area and protein on its own, and then proteins had to form in another area on their own, and then they mix together to start having some kind of biological function, like all these pieces just mixed together. That could have happened, though I would say that would be like my least confident scenario or hypothesis. What is more likely to happen is that one of these molecules, and what's really been shown, David Deemer's, like I point to his paper a lot, but his overall thing really showed that all you need is one biomolecule to form either RNA, DNA, a protein, a carbohydrate, or a lipid, right? But
But in and of
typically
itself, these one protein's not gonna do nothing.
it's proteins or nucleic acids like RNA, right? Those are the two that are really focused on, because those are the two that have function to build other structures and stuff in the modern day. So those are the two that are really focused on. And David Deemers showed that once RNA comes about, it's encapsulated in these lipid vesicles, be it phospholipids or a monolayer, bilayer, be that phospholipid or not, whatever kind of lipid vesicle that it's encased in, they're self-replicating, and so they will replicate themselves, and as they do, they have errors, or they have different nucleotides in their sequence from each other, and that leads to different chemical attractions, which eventually lead to a different biological function.
Okay, but
And
giving you all
so
that, okay, I'm
that
gonna
is
give you
how the
everything.
further
I'm trying to make it quicker if you
structures.
say you have to leave, so let's just try to simplify and make our statement shorter.
Well,
Because
that's
I'm
how
doing it.
the further structures get built,
I
right?
got
From
you.
one. You don't need them all being
Right.
built individually. Yeah, that's not
Gotcha. Okay,
the
so
most
giving you
likely
that,
way.
let me just concede all of that just for the sake of moving forward in the argument. Then what? You have RNA that's hypothetically self-reproducing. Okay. Then what? You'd have to start interacting with something to carry out its code. So
Well,
RNA is just like a code, genetic code. So then what? We'd have to
no.
eventually have nucleotides come into play to make proteins, and then somehow miraculously we'd have to have a translation system between two languages. So like
No.
even if we did have this encapsulated RNA, we still need a continuous influx of nucleotides, and we need amino acids to come about and start making this like language system so RNA can actually do something, because in and of itself it means nothing. It's like a computer code on a piece of paper without a computer to carry out its function. You have to start interacting with other parts to move forward.
Yes, you need the other components, right? So once you have self-replicating RNA encapsulated in lipid vesicles, right? Again, now we're moving to like the next step, right? So how do we go from protocells to like the muddy ground in between a protocell and a living cell, right? So how do we go from protocell to this muddy ground in the middle before it's really a living cell, but it has other structures than just RNA
No, I
encapsulated
got
in
it.
a lipid vesicle. So that
So what's
there's
the
a
next
lot of
step?
nuance in there. David Deemers did he did the beginning portion of it where he allowed the RNA encapsulated vesicles to keep replicating, and he altered he saw just how in the environment that they came about, which sequences survived in the environment, some that were eliminated. Like he witnessed this kind of natural selection process. Then he would put an acid or a base in there and see, how that changed the natural selection process, like which sequences were now surviving as they self-replicated. But he also witnessed other structures being formed in these protocells. So the start of something like a protein being built, we can't say for sure that's what it was because he didn't let the experiment go that long. But I'm sure other people, I'm not as informed about this part of the process as I am others, but I'm sure there's people who have looked into, how to go from these protocells with an original biomolecule to having other structures in the
Okay,
cells.
so going back to David Deemer's paper, he used nucleotides already. What he did find was that he had these nucleotides, got them to bind to one another, okay? And then he introduced lipids to get them to be self-encapsulated. However, he did not create the nucleotides themselves, or he did not create an RNA that was self-replicating in and of itself. I remember seeing this
Well,
paper. Yeah.
it's not just one paper, right? He wrote a series, his group wrote a series of papers. It's like 10 of them
Yeah, I'm just like
on this
compiling his work.
experiment that they did. And so you're talking about one part of the
Hey,
process.
did he make nucleotides?
Yes,
No.
yes.
Did,
He
did,
they
yeah, he made
formed them.
nucleotides. He made activated nucleotides like from precursor or chemicals. Is that what you're saying? Or didn't he use already like present nucleotides? Didn't he just introduce those?
I'm sure he did the experiment both ways, but I
I
know
can't find that he did that, by the way. And also, did he get a self-replicating RNA from his RNA polymers?
yes, he
It's
was
okay.
like the first one to report that these RNA encapsulated vesicles had behavior similar to biological evolution or natural selection.
Yeah, by doing the wet
So
dry cycles and finding that
they're
other.
self-replicating.
No,
And
the RNA. He did
I
not
do
do
have to go
self-replicating
though,
RNA.
so we can look into the specifics of that
Yeah, let's do it.
for next time. He's not the only one though, even, and I'm almost certain that he did do the precursor studies too, and they're in one of his earlier papers in the series. But if not him, there's other people who have done these prebiotic plausible RNA formation mechanisms. Just Google them. But we could look into what Deemers did for next time.
I'm looking into it now.
Re-
He did not make self-replicating RNA, and
Hold on,
he used nucleotides.
real
He
quick, you
basically got
guys.
them to
Like I
polymerize.
said, he has like 10 papers, if not more, about this experiment. So you got to look, we'd have to find the one pertaining to that step, which would be earlier in the process. So,
Real
Do you
quick,
claim that he's done that?
before you guys conclude, because this is exactly how this ended last time, with you guys going over specific papers for the debate, but then that never happened. Jayace, could you please accept Taylor's DM request, like her friend request, so then that
yeah,
way
I
you guys
did.
can go over. Okay, cool. So for next time, just can you guys have like specific papers for people to go over, and then we can get into the specifics.
Yeah, I looked at all the papers that he's ever posted that were pertaining to Original Life in the chat. It was like 40 of them, and yeah. So I just wanted to say that I did look through, even though he didn't DM me any. But that's why I'm trying to talk to him about him, like what they
Yeah.
did and did not contain.
Yeah,
So
you
maybe
guys
next
can.
time he'll check over his papers and be able to
And do you
confirm
have
that.
time for
Yeah.
question and answer or Q&A, Jace, or do you have to run? That was my next thing I wanted to mention.
Yeah, I do have to take off. Man, I'm sorry.
It's all good.
Can we
Yeah.
schedule another talk
Yeah, probably maybe around the same time next weekend.
next Sunday at 4?
Yeah, if you want. I mean, if you pick a paper that you want to talk about, right, send it to me. I accepted your DM, your friend request or whatever, so you should be able to send me stuff now or whatever. So send me whatever paper you want to discuss and I'll read it, and then we can either schedule a time, or if we just catch each other during the week, we could talk about it then. Doesn't really matter to me.
Okay, yeah,
Like I
that
said,
would work.
like I said,
Maybe we'll just say next Sunday at the same time.
yeah,
Would that
yeah,
work?
yeah. Like I said,
Okay.
trying to schedule things with me right now because of this other stuff I got going on. It's just going to be unreliable. So that it would probably be just if you catch me during the week, that would be fine. Or we could shoot for next Sunday at 4.
Okay, that'd be cool. Yeah, we can try to discuss particulars. That'd be great. Yeah, I have some more things I like to talk about with this, and I thank you for showing me this paper about the David Deemer's paper. That's really interesting that he got him to be encapsulated and have like some sort of proto structure in the cell. So I really appreciate you showing me that, and I look forward to talking with you again.
Yeah, same here. Thanks for chatting. Had a good time, and yeah, I'll see you hopefully next weekend.
Okay, looking forward to it.
All right, see you
I can
guys.
stay for Q&A if anybody had a question for me, but I guess he's have to head out. Biascles, was there any specific questions that were for me? Our Biascles drop. Oh, here you are.
No, not any specific questions for you. I really wanted. I mean, you guys are going on dating and radiocarbon dating for a while. And I wanted
Yeah, I know.
I wanted to ask about polonium halos that were basically formed instantly in the
Mm-hmm.
granites.
Oh, wow, that's a good point. Maybe next Sunday. Like, I was trying to schedule a time with him that he'd have like plenty of time. That's why I told him to pick, and then this was the time he picked, so I figured it'd be good. But
I think
then after we talked about radiocarbon dating, he said he would leave. So I don't know. I'm
I think
sorry.
you guys should just now that you guys have DMs and all that worked out, because you guys literally left off at the same exact point you did last time with the metonite clay or whatever. And then
Yeah, I know. I'm
going
trying
with
to
the
move
specific
forward, but
papers. Yeah, that was
yeah,
frustrating.
I'm so
Jeremy,
sorry.
I'm sorry, bro. I don't know what experiment you were conducting, but maybe you should have let people know about it.
I'll tell you later.
Well, next Sunday he should be here, hopefully. I've been trying to catch him because like he's very passionate about this apparently, on Discord. So, yeah, hopefully next Sunday we can finish up the convo. It does seem like a very slow progression. I try to move forward, but I think like, I was trying to tell him like maybe summarize your statements so we can because I try to make mine really short and concise. But if you summarize them, you can move on much quicker. So I was trying to get him to do that, but everybody's a little different.
All right, well, good discussion, you guys. Should we move down, Gio?
Yeah, we can pop. Yeah, is it ready to do that?
Yeah, I didn't see any questions in the chat or anything.
We can continue it there, but yeah, we'll do that real quick. Yeah, just pop back down to open it up, and yeah, we can
Can
I
continue.
do it? Can I do the move?
Do it, Gally.
Yeah, well, I already got. Yeah, if we do it like twice, it goes faster. So yeah, do it if you want to.
I could not figure out how to do the move. The channel names weren't popping up. I typed /move, and
It's
usually
F
like I
move,
can.
F move / F move.
Oh, all right. Next time
Yeah.
I'll get it moving.
Yeah, I was trying to progress the conversation forward like the best I could, but I think like he has like he speaks in more like long drawn out statements, so it takes so long to get anywhere. Ugh.
He does it till he's out of breath. I noticed he's like out of breath by the time, but he's still like trying to force out more words.
I know, and I'm trying to like interject and move it forward, but it's like pulling teeth, dude.
Oh, that's why I was saying you guys should for sure go on specifics with each paper. Like
Well, he just lied about his dude. So yeah, if he would send me whatever papers, like that'd be great. I'd love to look at them.
What do you mean he lied?
That guy David did not make it self-replicating RNA. Period. He didn't do that. That's just not what he did. You can even look him up and look up what different papers and research he's done and find that he didn't do that. It's not very difficult.
See, that's what I'm saying. Instead of saying like lie, like if you guys had the paper in front of each other and like the audience can have the paper exactly, and then you can
That's
just
true.
go
I was
over
trying
it.
to get in touch with him beforehand.
Yeah, so he said he'll do it for next time, I hope.
Yeah, that'd be cool. And man, Piezo DM'd me this really good point about dating that I should have brought up. Like, if he was basically saying there would be nothing to disprove that these dinosaur bones with soft tissue would like basically not be able to survive. But if we did do carbon-14 dating and we know that the rate of carbon-14 decay, then we would be able to say, okay, well, well, there's no way we can actually have this, like, in this, like, it should have, like, no carbon-14 whatsoever. Like, if it was actually that old. I don't know. Talk to Piezo. He had a really good point about that. But I think the dating conversation is a really cool one, too, in terms of evolution, because, you know, if he's talking about dinosaurs and he does work in this study, like, I thought that'd be interesting to talk about, like, some of the inaccuracies, because this goes on with the evolutionary theory. So, anyway, but next time we'll try to keep more concise and short. And
Yeah, you would just carbon 14 date that. It's organic matter. You would just date it.
Mhm.
Well, we know it's too old. You don't know jack shit. You made some good
I
points
know,
about the
right?
dating.
I'm glad that he finally admitted. The only thing I was trying to get him to admit was that there's assumptions. There's many assumptions made in the dating method. Whether you assume the rock age is this or this is that. I understand he's saying, oh, well, it's not assumed because there's like a constant decay rate. But like you're assuming a constant decay rate. Like you're having to assume a bunch, like something somewhere, right? So getting him to admit that, I feel like it was a big win at the end. So, and then getting to him to admit that the waters were muddied between like how we have this RNA and then what comes next. So, and I was like, okay, it just like giving it to you that we have a self-replicating RNA. What is the next step? How do we get to a cell? And he admitted the waters are muddy, or basically he couldn't give an answer. So I thought that was good too. That's why I wasn't trying to be too hard on him. And I want him to come back. That's the main thing. I want to keep talking to him, you know, since he's such a big proponent of this. Like, so I wasn't trying to be too hard on him. But then again, I was trying to like kind of move forward at the same time. So
He said the word right 786 times throughout all that. You guys weren't counting?
Nah, I don't know.
Yeah, I'm sorry if the combo was like a, you know,
You're
y'all feel like
good.
you didn't get much out of it. I'm just trying to move forward.
Left Lane just posted an article about RNA that evolves on its own. Hey, Left Lane, could you say exactly what you were saying in chat? You were saying that Taylor is wrong about many things, but couldn't name one of them. Do you want to share that now? No, just gonna shit in the bed then. He said you need to study a little more.
Then
That's
it's just
What?
then it's just heckling. Then is all he's doing is just heckling.
Yep.
He's a
Side troll
heckler.
heckling.
Call
Hey, if
that
I was
chat
wrong, that's fine.
balls.
Just tell me
In
what I was wrong
chat, he's
about.
got like very bold chat balls, but in voice chat, he's just not saying anything.
Loud in
Yeah, I
the
want to
chat,
learn. If I
but in
ever
the
am wrong, I
PC,
want to learn
quiet
from it.
like
So
a
please
cat.
tell me.
Well, I just think it's very unfair to say that she needs to study a little more and that she's wrong in many arguments and then not literally specify anything. Just like, oh, many things. Like what?
Well, next time I hear Left Lane talking, I'll ask him about it.
Oh, he's so bold in the chat. He just posted a paper. Here you go, Taylor. Check out the paper he posted.
Well, he might be doing something where he can only type and not speak vocally. Is that the case, Left Lane?
Well,
Link
here.
Oh,
try
What's his safe word?
I've actually been talking, but evidently you weren't hearing me, so I don't know.
No, you were muted. You had your
You were
mic
muted.
muted.
So what was she wrong about specifically?
I'm on push to talk. Like talking about the activated and deactivated nucleotides in specific. I mean, if you deactivate them, they're inactive. They don't work, right?
Yeah, they sure don't. So how do we get activated nucleotides? It's unfair for them to start with
Well,
activated nucleotides.
well, no, that goes to the step prior to. That's what Jace kept
Yeah,
talking. It's
they
not
know
all
to do
done in one
it.
step. It's done in many steps. He's talking about they had like 10 different papers, and y'all were talking about one portion of it, and there's maybe 8 other things that you haven't, that you're not considering. I would go back to the
Yeah,
first
different papers,
paper
different
and start
conditions.
from there, and maybe that's got answers. If you're starting at the end paper, maybe you should go back to the first one, and that might hold the answers.
So what should I what should I conclude if I look at all these different papers in each individual step and they all have different conditions and requirements?
Well, I don't know. You're making an assumption there.
No,
Well,
it would
that's
have to happen if life formed in this manner. It would have to be one step preceding the next. You'd have to have nucleotides formed to make RNA.
How do you know that?
Logically. Because RNA is made of nucleotides.
I mean, but see, you're making all these assumptions as if you know
What?
how it happened or how it didn't happen.
Dude, you'd have to have nucleotide synthesis before RNA synthesis because RNA is quite literally made of nucleotides.
Okay, so what do the conditions need to be for that to happen?
For which one? The RNA synthesis?
Yeah, any of it, nucleotide synthesis, any of it.
Well, there's actually a couple different ways, all of which have also different conditions. But we can pull up John Sutherland's paper.
The thing is, it's like
Um,
the point you're making, Taylor, from what I gather, is that there's all these different puzzle pieces that are in these different experiments, different steps, different papers, but when you try to put it all together to make a complete picture, it doesn't work. Correct?
So there are pieces missing. So there are pieces missing. Nobody argues that. It's not. There's not an exact conclusion on this. It's under investigation, and that's about it.
Lefley, I'd like to ask you a question. What assumptions, if any, is Taylor making that you think are unjustified or unreasonable?
She's arguing about the conditions that this had
Yeah,
to
let me
be
read this.
in to form, and she doesn't even know what the conditions need
Yeah,
to
here we
be.
go. I'm ready to read it. Here's the materials and methods. Dry heating reactions of TAP with ribose. Stock solutions of 2,4,6 triaminopyrate and D-ribose, which that's a big problem because ribose itself is really hard to make. So the fact that they started with this is poses problems. Were prepared in nanopure H2O. When dried at 45 to 95 degrees Celsius, an Eppendorf tube with 1 milliliter stock solution was placed in a dry block heater at the specific temperature. For the drying portion, they used 35 degrees Celsius. Tubes were placed in a vacuum centrifuge. I don't know where that would be on early Earth either. Samples that were reacted for multiple days were then dried for 24 hours. The resultant high viscous solution was momentarily resuspended in the original volume of nanopure H2O and nanopure water, and the process was repeated for an indicated number of days. So they predetermined how long they were going to do it. Momentary hydration was performed to ensure the solution was homogeneous during the course of the reaction. Additionally, daily hydration of the reaction mixtures model environmental day and night cycles. So these wet and dry cycles that they're having to invoke. A process which had been operated in prebiotic earth. And HPLC of samples was performed by running 9%, 0.1 molar NH4 HCO3 and a pH of 7.2 and 5% HCH3OH at 0.5 milliliters through a Phenomenex Sindri Polar RP 80 column.
So
then,
I mean,
yeah, so you
it's
can
like
see how many different conditions they even had throughout their own experiment.
but no layman has any idea what you just read.
You asked.
Well, okay, what is your critique of what you just read?
Well, first of all, they're having to continually add and change the conditions. They're having to like centrifuge it. They're having to use a vacuum centrifuge. They dried it. I guess the wet and dry cycles could be explained on a wet and dry cycle on the Earth. They gave it the proper pH and inputs, and yeah, they were just basically priming it with all the right conditions. This is for one paper. And then so now if I go look at a paper that this one was for the synthesis of the nucleotides in particular. Okay. So now we'd have to compare this to the materials and methods and conditions, like right here, pH 7.2. So I need to compare that to what is the pH of the next step. Is it compatible? Yes or no? The temperatures that they dried them at. The wet and dry cycles, would it constantly need to be in a hydrogenous solution? Would it need to be like hydrated, or would it need a dry cycle continuously? Because some biopolymers require different conditions to be stable. So that's the thing. Like, so it's not just this in and of itself. It's this compared to another preceding step that would need to take place.
But how do you justify making that statement? Have you done this?
What?
What?
Have you done this? How can you justify saying they've done something wrong that you haven't done yourself?
Okay, so you think the only people that can talk about original life researchers isn't people who actually read the papers, which is the purpose of writing a paper, so other people can read it and understand it. You think only the people that do them can talk about them?
No, that's written for scientists that have done it and understand what I have no idea.
This is written
I have
for all
no idea what
scientists
you're
to learn
reading.
about it.
But I would posit this, Taylor. It's just the fact that you're critiquing it, you can question everything you want to. And how the heck does that happen? But you critiquing it or saying it is wrong, you're not in the position to do that.
Hold on, you
Hang
don't
on,
know
real
why
quick,
you're saying
real
it
quick. Let
wrong.
me just interject.
Anybody's in
Left
a
Lane,
position
why,
to do that.
Left Lane, why is Jayace more qualified to talk about these things than Taylor?
He hasn't done them either.
He has done these things. He's a practicing scientist.
He's
No,
got a
I'm a
license to
practicing
think.
scientist too. What do you mean?
Are
Left
you
Lane gives that
employed as a
license.
scientist, Taylor, or are you a student?
Yes. I'm employed to do research. He's a grad student too. He's at the same level as I am.
Okay,
Yeah,
So
they're
so
exactly
I
the
would
haven't seen her.
same,
assume
Left
You
Lane.
haven't seen her.
I would assume that. Wait,
Left Lane, do you
wait one
understand
minute.
they're
Let
exactly
me
in the
let
same
me
position,
interact.
Left Lane? Do you
Let
understand
me
that?
interact
They're
with
the
her.
same?
Let me
You haven't
interact.
seen her Twitter
on,
account
wait.
yet.
Holy.
Wait, guys, wait. One at a time. Left Lane, now do you concede the fact that they're on the same professional level so you can stop appealing to
No,
credentials?
no, I don't know. I would need to see a paper. I'm sure she's employed.
What?
She's written a paper. She's written a dissertation of some sort. She's done some kind of
Hold on.
Where's
complementary
hold on,
research, some sort of complementary research that would validate what she just said.
So you're
Okay,
just
Left Lane, the thing is,
you're
at
saying that
best,
I'm being honest. Left Lane. Is that what you're saying? And would
At
you
best,
have you
Left
asked
Too many
Lane.
people
Jace's
talking.
papers?
All
Have
right,
you
guys,
asked to see Jace's papers yet?
Jayace
Yes,
doesn't have a
yes,
paper.
I've seen
we don't
Jayace's
have
work.
a paper. We don't have a paper from Jayace. Let's see it.
He has written papers. I've seen the
Post it.
papers.
Where?
Post it.
Link them.
I'm not in possession of his papers, but I've seen them.
Post it. You ain't
Okay,
seen
so
shit.
About what? What has he written a paper about, Left Lane?
Oh Lordy, I don't know what it was about.
this is
He
ridiculous.
has published. He's published several papers.
Post it. Post it.
Okay,
or
so
you're
let's
bluffing.
get this
I don't have
straight. 5
Jace's
minutes
papers.
ago,
You're bluffing.
you thought that Taylor wasn't qualified to talk about this stuff when she has the same qualifications at Jayace. So you were just
I
full of
disagree
shit.
with that. I
And now
disagree
you
with
were
that.
hang on. I'm not done talking. I didn't interrupt you. You were just full of shit. Now you're saying that Jayace has written papers, and we're just supposed to believe you, even though 5 minutes ago you just talked out of your ass. You're just full of
No,
shit.
yeah. See, that's all you do is
He's
just say you
assuming
talk
that I
out of your
Have we
ass,
got
and you're just
Jace's full
stupid, and
name?
all you do is
Left Lane.
talk out of your
Left Lane.
ass.
No, I'm
He
not
just
calling you
assumed
stupid,
everything.
but you've accused Taylor of being wrong. You said on a professional level, students aren't scientists. She's a scientist. She's not a student. You'd said, I'd sincerely be interested in following her progress. She's arguing against the folks. She should be convincing. She'll need a few years of study for that. Again, they have the same qualifications. She needs to study a little more. Getting stuff wrong isn't an argument. So, Left Lane, I want to say to you, getting stuff wrong isn't an argument. You're full of shit. You're
This is
just
silly.
wrong.
I'm not going to talk to you about it because you're not
Hold on,
qualified either.
Left Lane, hold on, Left Lane. I've asked Jace for papers. He hasn't given none because he doesn't want to get doxxed. How did you get them?
Yeah.
I have seen Jason's work. We communicate.
Okay,
He did
permitted.
it. So
Well,
We're supposed
percentage.
to take your word
you
for it,
do
but you
know.
won't take mine.
You do know that he's got papers because he didn't want to post them here and dox himself, right?
Percentage.
What's his full name?
Okay,
Left
so
Lane?
you're assuming that I don't have papers first of all, and you're saying that I'm dishonest about my credentials.
I didn't
However,
say you
we're
did. I
supposed
did
to
not
just
say
hold
that.
on.
I did not
I'm
say
still
that.
talking. I'm
Yeah,
still
but you
talking.
said something
But
I did not
however,
say.
we're supposed to take your word for it that Jayace has these papers, and somehow that would make him more
Take his
qualified
word
to have
for
this
it.
discussion than I do.
Ask
Than I
him.
am.
Ask him when he comes in. Take his word for it.
Yeah,
I'd
well,
like
I
to.
can say I have
at
paper
best,
doesn't
your
mean that could.
position is appeal to incredulity. And at best, your position is appeal to credentials. That's your best position.
All
Left
right,
Lane.
y'all go.
That's some hot garbage, man.
Y'all go ahead. Proceed with your conversation.
Left
Yep,
Lane,
but
go
it's
through
so
her
funny you
Twitter
say I'm not a
account. She
practicing
posted
scientist.
stuff up there if you want to go check stuff out.
You obviously don't know what you're talking about, Left Lane.
I'd like to
Did you not
reset-
even know that me and Jace were on the same level academically? Did you know
No,
that?
you're
when you
not.
made
No,
those
you're
statements?
not. No, you're not.
No, I'm okay. So I'm lying.
They
Is that what you're saying?
established that at the start of the debate, Left Lane. At the start of the discussion, they established that. You might have missed it.
No, I
But
heard
they
Taylor say
established
talk
that.
about being a fellow scientist, but yeah, it's the first I've heard of that part. But
They
yeah, I
established
don't
that
know.
they're
I
on
disagree.
the same
The,
page.
Well, then you're
the
in
line
the
of
debate.
the line of questioning and inaccuracies in her presuppositional
What?
ideas and what they got wrong and
What was
Jace
inaccurate?
correcting her and
Yeah,
then
name
her
one thing,
referring
please.
to the same thing. that indicates to me that she's not saturated in
Where did I get
knowledge
inaccurate?
on this
All
topic.
right, yeah. Name one
Where did I
thing
get
that's
inaccurate?
not wrong or inaccurate, or else you're full of shit as usual. Name one thing.
Just go re-listen to it. Is all I can tell you.
Oh, you can't even give
Wow.
an example.
Get
This
the
is so
fuck
funny.
out.
I hope
Yeah,
y'all
you
He's
hear
are,
this,
being
dude.
he's being inflammatory.
you are the most
You said that I was
disingenuous,
wrong. Why did
dishonest
you say
person,
that
man.
if you don't have an example?
All right, guys. All right, guys. All right. Let's reset. And for the layman's out there, as Left Lane said, what does that even mean? All right, for the layman's, it's pretty simple. You take one experiment, one paper, and one type of condition, right? All these conditions that are required for one of these steps, okay? What she's doing is comparing and contrasting the different conditions for the next preceding step. It's very simple. When you compare and contrast other papers, like the preceding step, going from like nucleotides to RNA, the conditions are completely different. Hence, the puzzle pieces do not fit together to make a complete cohesive story of abiogenesis being valid. That's the point.
100%. 100%. But I really want to know what I was inaccurate about, Left Lane. If you comment and typed
He's
in the chat,
deafened. He's deafened and muted.
wow. So he's just gonna
That's-
cop out. Seriously.
That's
Yeah,
all
This
he
is
does,
hilarious.
Taylor. He
just
he
shits the
does
bed.
that.
That's
He
right.
heckles,
Yeah.
and then he doesn't defend what he's heckling. That's his whole thing. That's all he does. He white knights, he heckles,
Seriously.
and you ask him for specificity, and he's just gonna run away. Just go re-listen to it. Okay, it was a discussion.
He was
Yeah.
scrambling by the end.
You can't even give me a freaking example.
You don't want to, yeah. I mean, if there's anybody that actually has a question about what Taylor is putting forth, please just, you guys could have a good faith argument if you want to actually understand where she's coming from. I've personally listened to the Abiogenesis presentation from her and Dan, and then listened to the Grayson and her debate, and I have a somewhat layman's understanding, but I have a a good understanding about like what's going on, what she's putting forth, and essentially it's just these puzzle pieces don't fit together, and she's just comparing the different conditions that were required for each of these steps. That's it. I mean, if anyone has a question about that and in good faith, we can just move on from the hecklers.
I always ask if anybody has any questions, and for my presentation, I particularly said I want to hear any rebuttals. I'm always open to anybody refuting anything I ever say. I would love to correct anything I say wrong. But to just say, "Oh, she's lying," and then not give me an example is so frustrating. Then why won't he
I
debate
wouldn't
me
let a
then?
truck
If he knows
driver get
better
you
than I
upset.
do to say I'm wrong,
He's-
then tell Left Lane to come debate me on stage.
Nah, he deafened. He doesn't want to.
You're
Whatever.
gonna say
Yeah,
he's not.
fanboy.
You're not qualified to tell him it was right, but he's not even qualified to say to ask to tell anybody that they're qualified.
Yeah,
It's
he's
just
not
so
qualified to say that
retarded.
I'm
'Cause even if
incorrect.
he's right, it's just an appeal to credentials and appeal to incredulity. So best case scenario, his argument's fallacious. It's so retarded. Like, do you guys get that? Even if he's right about everything he says, it's still an appeal to credentials, appeal to incredulity. It's not an actual argument. It's like best case scenario, his argument's fallacious.
Does anyone actually
Dude,
have
I
a
actually
rebuttal?
got pictures. Well, I don't got pictures. She got pictures on X of her being in the lab.
Yeah, I live stream me working in the lab. Like I have nothing to like.
Yeah,
I'm
you're
always
good.
open
Your
about that I'm
credentials
doing
are
this.
solid. Your
I
credentials
would
are
rather
solid.
not dox myself, just like Jace. I get that, so I'm not gonna say that he's not like he doesn't have qualifications to talk about it just because he won't post his papers. Like that's fine, but for him to be like, "Oh, I'm not qualified," and he obviously doesn't even know what I do, and then how would he be qualified to even say I'm wrong at that same rate? If I'm not qualified to say anything about it because I haven't physically worked on it, then he's definitely not qualified to tell me that I'm wrong. And even if he was, then you need to give me an example of what I'm wrong about.
All right, so questions on the thing. Let me see. So, like, if I had the flour, the cake, the eggs, the icing, everything in the oven, everything to make a cake, I could make a cake. If I had the seed, the water, the sunlight, I could make the tree grow. Why do you think it's impossible to grow a human with all the ingredients?
Look, you're talking about a tree growing. You're using
Yeah, and
already
other things.
existing life.
I mean, I could. All right, plastic. If I had plastic, a heater. I could get a chair. I can make a fire. I can get a car.
You
If I
do
got all
it.
if I got all the parts, I can make these things. Why do you think it's not possible to make a human?
Because all those parts that would have to be synthesized in the same condition require different conditions, quite frankly.
No,
So
I mean, if
you'd
we
have
get
to
them,
have some
get all the
mechanism of
parts.
how that'd be functional. And even if then, like that's what I said, I even gave Jace the whole point. I said, okay, I'm gonna concede everything. Like, yeah, we can hypothetically get this self-replicating RNA that's encapsulated. Okay, even though they didn't start from prebiotic conditions, they didn't start from scratch in this experiment in the guy that he referenced both times that we spoke. So even if I did give him that, what is the next step? He was not able to give me that next step
No, I mean,
whatsoever.
no. But
Because then you'd have
I'm
to have
asking you, why do you think it's not possible? Given if you're given all the material to make something, why do you think you can't make it? What's
okay,
the missing ingredient then?
there's no missing ingredient. It's the fact that we don't have enzymes to make these reactions happen, and you have to use different conditions because they're completely different types of molecules, and they require completely different conditions to make these things bond in the absence of an enzyme.
To make
Unless
it,
you can
The
demonstrate
missing
it.
ingredient. Oh, I thought you were done.
No, you're good.
The missing ingredient is information. You can't just take inanimate matter and all of a sudden there's some kind of information there. Information is there's a code in the DNA. You have to have a sender and a receiver, and they have to agree upon a code, right? Doesn't this all work by the DNA tells the thing how to build and how to what to do and shit?
Yeah, and it builds the DNA. So that's another point that I was about to mention, which is like, even if we did have everything, like we need systems already in place, like translation systems, and something to carry out a code for anything to function. So, like for example, we have 2 language models in the cell. We technically have 3 because the RNA has 1 different nucleotide than DNA, but that's just a side point. But so we definitely have 2 completely different language models, one for the proteins and one for the DNA. The DNA gives the code to the proteins. There's a system called translation where they will translate the DNA code into proteins. So
yeah, I mean, that's all true. I'm just saying, given all the DNA, all the nucleotides, all the ribosomes, give me all the stuff. If I give you all the stuff, you still, what's missing? I mean, just the accounting of where you're just
I mean,
saying, well, you can't account for where
what
the
Jeremy
information
said
came
was a
from.
good point. What's missing is the information to know what to do with it. This is inanimate chemicals. They have no end in mind. They don't know anything. They're just responding as the environmental, factors would allow. So they're just constantly bonding with other things, and so it's not going to be anything cohesive. And then so even if you do have everything happen to bind in the right way in different conditions and they come together, like moving past the incompatibilities in the chemicals, then what? They have no information or end in mind. You have to have a DNA code form that can actually give like proteins that are functional that can
Yeah, but
actually then go and
we
replicate the
do have
DNA.
that information. We have it.
Where? From where? Where to get the DNA code from?
Yeah, you're just asking how do you account for where it came from? I'm just saying, given all the information, if they're not worried about justifying where it came from, they're just saying, okay, I just stole it from a baby and I stole it from a
No, dude, hold
here.
on, hold
Why
on,
can't you
James.
make it?
No pre abiogenesis, no information, all inanimate matter. In order to have that step happen, you need to have the inanimate matter somehow get this information and be sending and receiving it. Like,
And multiple systems already in place.
It's a
Well,
chicken egg situation.
people will just say that the universe is fundamentally understandable just because it's a brute fact
Okay, let me
that it's
just say.
understandable.
Okay, well, let me just say.
You can
Okay,
get
we
the
have a.
information because it's just it's intelligible. It's intelligible because it has information. I mean, there's just
Okay, that's like me saying we have like a bunch of metal scraps here. Okay, a bunch of metal scraps. Just for the sake of argument, we're just going to add like hydrochloric acid and then say that they can form different, shapes and stuff. And then over time, like, why wouldn't that be able to form a functional computer? That's exactly what it's like saying.
no, it's not. It's
Why?
it's just the chemical reaction that already exists. You're just asking for an accounting of how the information exists, or it
No, I'm
exists.
saying if we
They
have
have
all the
all the
metals here and we're constantly like melting them and cooling them in different conditions, like, wouldn't those metals just be able to form a functional computer theoretically?
I mean, where do you draw the line? I mean, what I'm saying is they are performing a function. They are the information. It's it. Yeah, the
You're
information's
not
there.
answering.
Yeah, I'm not asking you to form it randomly. I'm just saying, yeah. I mean, if you want to account for what the mechanism that drives like evolution and the accounting for that, that's one thing. I'm asking, if these guys get all the materials, they take it from a baby, the girl, and a baby boy, and they make a baby in a lab. I mean, what are you going to say?
You're using already living cells. You have to think about inanimate materials. That's what we're working with. So that's the same thing like saying a computer can form from melting metals and cooling them multiple times. Even in one of them's hotter, one of them's colder, different shapes. Over time, eventually you'll get a computer functional just from melting metals and cooling them. You have
Yeah, if you
everything
have all
there.
the materials, from a computer, you can't assemble those anyway. You need to have the software for it. You know what I'm saying? Like just
But
having the metals and all the physical stuff doesn't really, you don't get a functioning computer out of that
yeah, I mean,
unless
So
you
where
have
does the
I
that.
computer code come from?
Yeah, I mean, so it's just a kind of where I understand like top down versus bottom up. That the evolutionist believes in bottom up, where I would say top down is more reasonable. That these forms, these things are, we have them. There's a difference between material and form. Physical and corporeal. We have the form, we have the whole, and now we need the parts to make up the whole. It's all top down. I understand that, but they're just going to eventually say, listen, I combined all these materials and I made it, and we're sitting there, we're sitting around saying, well, that shouldn't be possible. Why would
That's
that not be
intelligent
possible?
design, though. If you're saying, hey, I combined all these materials and made it. Hey, look at that, intelligent design.
Yeah, at the end
Well,
of
I
this,
mean,
what do
they'll
you
just say
prove?
no.
At the end of this, what do you prove? It took intelligent design to make something? I know.
Exactly.
They're gonna be like, oh shit, we did it. Even if you could make life, they're gonna be like, oh shit, we did it. We made life. What now? What?
Hey, way to
Yeah,
prove intelligent design. Nice.
well, that's what I mean. Yeah, all right. So I mean, if that's just all we're trying to argue, I'm just asking. That's why I was just asking. All right, or do you hold the belief that's even possible, given all the materials and the information that you could make a person?
Information comes from intelligence, complex, specific information.
I'm not arguing that.
There's different types of information, but anything that's complex and specific, we see from intelligent designers.
Well, I think No Tanks is going into more of the metaphysical argument. Like, even if you had all the material composition to make up a human, would that still be a human? My philosophical bias would say no, because I think there's more to what makes people people than just material. I'm not a materialist at heart. Like, I think there's a
Any
soul.
living thing, any
Yeah.
living thing, dude. You need the information. The information has to be there. Information is not physical.
Well, yeah.
Like information isn't
Physical.
physical. You can't paint. There's a good thing with cathexis and QE. He goes through it. There was also a debate after that thing that he gave, but I can't find a debate. It was with Robert Bassano and a couple other good guys. But yeah, the information you can't you can't. It's not a physical thing anywhere. It's like supernatural or metaphysical, whatever you want to call it. Like it's not. The information is not tangible. It's not
Yeah,
physical.
they do differentiate. I mean, if you go into the category, they do differentiate between material and physical. Physical could be material or energy. Energy being information.
This isn't any kind of physical thing that they're taught. Like there's no physicality to information, even in their idea where like light is physical or space time bending is physical. Like the information isn't even on that level. Like if I write a whole bunch of letters down on a piece of paper and hand it to you, you're not going to know what it is. If I give you a code to decipher, if me and you agree on a code and you now know the code, and I give you this, what appears to be jumbled up letters, now you have the information.
Yeah,
Before that, it
I
wasn't
agree.
there. You had all the physical stuff, you had the paper, the ink, you had it all, right?
Yeah, I
But
agree. That goes to
there
the
was no information there until you know the code. That's what I'm
yeah. That
saying.
was
It's not. There's no physicality to it at all.
yeah. That was kind of what I was referencing when I said top down, dealing with the whole rather than the parts. You need I can give you all the parts, all the letters, but you need the whole. You need the alphabet for it to make sense. Yeah, the
No,
primacy
you need
of the
more than the
whole.
alphabet, bro. You need a code. Well, yeah,
Well,
if we're just using English, then yeah, you need to know the alphabet and the code of how English works. But even if I give you English letters, you may still not understand it because then there may be another code layered into it. You know what I'm saying? It could be a code within a code. You never figure it out.
yeah, I don't know. I mean, they'll just appeal. I'm just saying, they'll just appeal to something as if like the universe is just fundamentally informative and intelligible and contains intelligence, and we're just a piece of that. They'll just appeal to that in some way. That's why I was just asking her, like, if they could do this, are you saying they couldn't do it because they would literally need God to intervene to give the creature, a sentient creature, like some kind of rationality or sentience or conscious? I don't know. Like, what's the missing ingredient?
Are you asking Taylor? I mean,
Yeah. Well,
we're
that's
not
what I
even
asked.
getting. We're not even getting to the. She's not even getting to the sentient life aspect. Like they can't even get past the RNA replication, and then go from there, and then
Yeah, and they can't even
make
get to
DNA.
RNA replication from nucleotides. Sorry to interrupt.
Right. Yeah. Exactly. No, I got it.
Yeah, I think I mentioned it before. There's like, I'm not one of them. I don't believe in evolution even a little bit, but like, adaptation, sure. But there are like theistic evolutionists who believe that evolution is true, but unguided evolution without a God would be like impossible. So, I mean, I don't know. That's why I was asking where you draw the line.
100%. Man, I thought he'd be like a really good challenging debater because like he works in the field in biology, and so I don't know. Everybody told me that he was like so passionate about it and posting all these papers. I feel like I haven't really gotten very far.
Taylor, you're saying they can't, like, I know they're doing all this in separate pots, but they can't even do the pot to get a nucleotide, you're saying?
Well, yeah, so they're talking about hypothetically having a self-replicating RNA. And this is what I brought up to him. And if he, never mind, but I brought this up to him, okay, and told them that like for these self-replicating RNAs that he's invoked as to be like the start of the first life form, like
That's not the
basically you would
start.
need an RNA. They cannot get RNA to self-replicate just from raw materials, just from nucleotides. They're having to
You need
insert
to get a
things like
nucleotide before you can even do all that, though.
yeah, and that's a
Can they
huge
do?
that's like
Can
one of
they
the
do?
hardest things.
Can they make it though? Can they make a nucleotide?
Mm, it's complex, but like so whenever they did make one, they had to use the ribose sugar. They had to insert ribose sugar, and then the ribose sugar would have a different condition to form. So they were able to make a nucleotide, but with already the ribose formed, which is actually really difficult to make, and they admit it. Like, so but if I was to say that to him, he'd be like, "Oh, well, they don't admit that." Yes, they do. They literally say that the ribose sugar backbone of the DNA is extremely complex and hard to make. They were able to make a ribose from prebiotic conditions in another experiment, which was like a huge breakthrough. That was in John Sutherland's work, which I mentioned. However, that's different than binding that with other things to make a nucleotide. And they're having to insert these activated um phosphorus chains basically to make it activated so it can actually bond. So
How
it's
far
very
can they?
unfair. It's very unfair research, and it's very very complex and hard to even describe one of these reactions. But no, they're not doing it in a one pot census.
I mean, how far can they get just with inanimate matter?
They can get one step at a time, one small step at a time. That's how far they can get.
You said
They
they were
never
using
like, for
sugar.
example, like they huh? Yeah, like a ribose sugar. That maybe that's as far. I mean, or you could just say if you believe in protein first, then they can get amino acids from like the Miller-Urey experiment, but they wouldn't then take those same amino acids for the next step because it was 98% tar. It's a lot. So they've been trying to do this for a long time now. They have no shortage of research on it. So to say, oh, well, it's not being funded or nobody's studying, that's so untrue. They've been trying
Yeah.
to figure this thing out forever.
Have you seen
And
the
know that like
Miller-Urey experiment,
yeah.
Jeremy?
Say it again.
Have you seen the Miller-Urey experiment? As far as they got, like she was saying, it's like 98% tar. This is what it looks like, bro. This is their most cited experiment for A-Biogenesis, and it's a dirty bong.
Dirty
That's
bone.
the.
I'm not even kidding, bro.
That's funny, but I don't know. You're saying they can make they can make so they can make an RNA from scratch.
No, they have not made an RNA from scratch from inorganic compounds.
Yeah.
They'll actually start with organic compounds and say, "Oh, well, we think this organic compound can form in this." in this way. So let me just tell y'all the difference. There's inorganic compounds and organic compounds. Inorganic compounds is like just what you find naturally in nature. They're not used for life. Organic compounds can be much more complex, and they are used in life. And so basically, the step from inorganic to organic compounds is like what the Miller-Urey experiment did, where they formed amino acids from methane and
Oh wait,
carbon dioxide and stuff.
how many atoms though? Right? How many atoms,
10.
Taylor? Yeah,
10
10
atoms.
atoms. What's the
Nothing,
most?
literally nothing.
So wait.
I mean, there's something, but
Oh, Mister, you're hot micing.
Sorry, dude.
So yeah, you said what? 10 atoms, 13 atoms, and then
yeah, 10 to 12
what's the
atoms.
what is the amount of atoms that is needed to get to the next step?
I mean, it's probably heavily debated, but you'd need like sequences of these amino acids to make a protein.
And the protein was what, 100 million atoms? Something like that?
A DNA is.
Oh, DNA.
But we can do the same thing with the RNA. How far have they gotten? You know, I think that they've just gotten to one nucleotide, but using like ribose. Like they're already using like the sugar backbone. So it's not fair.
Yeah, they cheated. Yeah, at every step
Yeah.
they cheated. Yeah, but I think there's a very specific reason why Jace doesn't want to go over specific papers for that exact reason. I mean, you guys literally ended on the exact same spot you ended off last time, and then said, "Hey, we should do a debate."
I went with his guy. Like his David, what's his last name? I can't remember his last name. David Deemer. Okay. So I'm looking through all David Deemer's work. This is his guy who we continued to mention this time and last time we talked. And so he didn't even know about David Deemer's work. David Deemer did not make a self-replicating RNA from nucleotides. By the way, nobody has. And also, he used nucleotides. So he just got these nucleotides to form, to bond together. He used nucleotides already primed and ready with activated, like phosphorus, right? So he put the phosphorus in there so they would be activated. So he completely started from unfair conditions, and then he got these nucleotides to bond just like they would naturally. They're already like in chemistry. If something is like primed to bond, like you have this phosphorus that's like needs another electron, for example, it's gonna bond. So basically they got this to happen, which is not that impressive, and then encapsulated it in a lipid that was introduced. Okay, and then they say this is a protocell. Are you kidding me? You literally got nucleotides to bond together for a quote unquote RNA that's not even functional, and then introduce lipids and say, oh, look, these lipids surrounded it. Like, that's his paper that he's citing. And then, like, he didn't even know about his work because I told him that they use nucleotides already, and he acts like he didn't know that. So then where do I go from there? I did look at his papers that he referenced last time, and he didn't know about them. So then what? I brought up his papers, and he didn't know about them. Like he did not know that they already use nucleotides, or he tried to dispute my claim that they did. Well, you would know that. Well, yeah, exactly. I mean, I'm not trying to be too hard on him, but that's frustrating. Like, if you're gonna give me a paper or a guide to reference, then at least know what they started with and used. I'm sorry, I'm venting.
That's too much to ask, Taylor. Come on.
If you're gonna accuse someone of being wrong, maybe with like specificity, it'd be good to bring up examples. Otherwise, keep your claptrap shut.
So, if there's anyone that's actually, because like this is so fascinating, this stuff, and I'm only interested because Taylor is so interested into it, and I was like, huh, what are they claiming? What are they doing with abiogenesis? Like, because I only knew the surface level stuff, like, hey, we came from rocks, and I was just like, that's retarded. Never looked into it more, and that's my own personal bias. But then Taylor with the specificity of why it's retarded is really interesting. I'm gonna post this for anyone who wants to actually like interested in it. There's this great playlist we watched called Debunking Chemical Evolution, Origin of Life and Abiogenesis. It's like a 6 video part series, and they're like 10-minute videos each. and it's really good. It's like with animations and everything like that. Yeah, I highly recommend it. I just posted it in live stream.
Yeah,
Mm.
thank you for everything
Yeah,
today,
so
Taylor.
not
Oh, go ahead.
I was just gonna answer his question. I just double checked. No, the nucleotides have not been made in a one power reaction, like the ribose and the nucleotide, which ribose is a part of the nucleotide. They'll introduce that already. So the answer to your question is no. And this is the basic subunit of RNA. So that's pretty sad.
Yeah, I feel bad for people who are actually arguing on the other side of it. Like, in all honesty, like, it's a hard argument. Like, if you had to actually stand by abiogenesis, you would be in. It'd be like 32-0 in a football game. Like, it'd be tough.
Well, then they need to not be like talking about it as if it's a matter of fact. That's the only problem I have with it. Like I'm telling you, like on these shows, like on like How the Universe Works, for example, it made me so mad. They said, "Oh, all you need some sugars and all you need is some amino acids, and boom, you got life." And they put this on TV, and people think it's true.
Yeah, it's totally misleading, Taylor?
Yeah,
It's like putting a cartoon ball on TV. Like, that's misleading.
yeah, I hate that. Mm. Were you gonna say something to me, taking back in?
Oh, yeah, thank you. I was looking at cell necrosis recently. It's pretty interesting. I was just wondering what the early cells must have been dealing with. I'm sure they would have been dealing with uh, cell breakdown. I've seen, like, for instance, I've seen how copper, how the electrical conductivity, if I'm not mistaken, helps penetrate or break down the cell wall. I just wonder how these early cells were able to survive.
Man, that we haven't even finished step 1. So getting to all these other problems is like I
Do you have
want
that
to, but
chart? Can you post that
there's
chart? You know what I'm talking
Mhm.
about, where that chart that says you are here? I think that's a great chart to
Yeah,
just
that's John Sutherland. He's made some of the most recent quote unquote progress as an original life. He's one of the best original life researchers, and he said we are here at the very beginning.
here. I'm gonna find that chart.
No, yeah. Let me see. I have it right here.
John Sutherland chart.
You're probably gonna see all kind of stuff that has nothing to do with this, but
You are here. Yeah, I want to save that.
Okay, here we go. Studies on the Origin of Life, the end of the beginning by Nature Reviews Chemistry, Volume 1. Here, I'll send it to you.
Okay, I'll post it.
It actually goes over other things too. There's so many issues, like where do you begin?
Wait till you guys see this chart.
Here, we just send this. There you go. It's sent. And then I added, okay, you can crop that, but I said I argue that we're way back there. I can move that out the way, but oh, it's embedded in there. I can paint over it real quick if you want me to.
Degree of aliveness. Inherently favored prebiotic chemistry. The current state of the field.
Yeah, right after that.
Energy-dissipated exploration of macromolecular sequence and composition of space. Major system innovation. Look at that jump.
How long, just curious, Taylor, has like abiogenesis been like a major field of study?
It was proposed
Since
like
Darwin.
100 years ago.
You know this.
70. The past 70 years they've been doing research, like field research, at least.
Yeah, since Darwin, dude.
Yeah,
Yeah, on
so
the
like
origin.
I brought this up the other day. Like it doesn't surprise me at all if it is true, which I'm not saying it is true, but like that they have, 50 years, 70 years to study something that happened 4 billion years ago, it wouldn't be surprising at all if they have gaps in their theory, right?
Well, it's not
I
just
wouldn't say
gaps.
I wouldn't say
It's
gaps.
like
I would
it's
say
a false premise.
inconsistencies.
It's a false premise to begin with that we came from fucking lightning soup.
Yeah, I study unicorns. There's no unicorns around yet, but it'll happen. Okay.
Appeal to time.
Well, yeah,
Or
but
appeal to the future. Appeal to the future is what it's called. Well, maybe one day. But
yeah, but
actually, I wouldn't say gaps. I would say inconsistencies, and I would say the more we study it, the further back we're getting. Because, like, if you look at this chart BS Quiz just posted, they thought whenever they came up with original life theory that it was extremely easy to solve. Like, okay, we just got these compounds, and boom, you got life. They had no clue of the complexities and requirements and integrated systems needed for the most minimal cell. They used to think that whenever they came up with this theory, I did a lot of research for my presentation on what did they actually think about the cell at this time. They thought it was a protoplasm that was surrounded by a membrane and had a nucleus in the middle. So they didn't even know about like the central dogma and stuff, like the information of DNA to proteins, DNA to RNA to proteins. They literally, this was published after they came up with the origin of life hypothesis. Like the fact that DNA has the information that then gets translated to proteins. That just shows like where they were at. And so at the beginning, they had really like high hopes for original life research
In
because they
layman
had no clue.
terms, the more they studied, the more complex it got and sent them further back in their research.
Yeah, progressively. To this day, in my opinion.
Like the better the microscopes got, the more complex these systems were, and those assumptions, those original assumptions, got thrown out the window. Hence, starting from square one every
But
time.
they'll never throw them out the window because that's all they have. If we have life, then we have to have some naturalistic explanation unless we want to invoke anything else. They would never do
They
that.
don't want to do because they're philosophically biased.
So they're
They're
gonna have to
creationists.
say,
Yeah.
they're left with no choice other than to say life formed from non-life. But now, how did that happen? Is the thorn in their what would that saying be?
Thorn in their side.
It sound like thorn in their side. It's been a problem.
so
Can't
would
figure
your
it out.
position be, and I'm interested in your answer too, Beasticles, that there can't be a naturalistic explanation for the origin of life? It has to be a intelligent designer.
I would say that based on the research that we've done, there is no plausible explanation.
I'll give
Yep.
you one. Morphic resonance, man. That alignment of the crystals seems to create information and order. So there's something going on there, especially with those
100%
oxygen
requires
transport.
intelligent design, and using the what is it, the 747 example. I'm sure you've heard of that before. Like it's like saying
No,
that
I haven't.
it's like saying that a 747 was basically formed by a tornado going through a scrapyard.
But with a million years,
Yeah.
Like
Yeah,
you
but
have a whole bunch of things. You got we have like in reality we have like trees, we have I don't know, we have clouds, we have just things, water, we have things in reality, and those things, those individual particulars are a whole system of their own, and without the goal or like the initiative to achieve that whole, the parts would be meaningless. Maybe
Yeah.
they wouldn't have any kind of goal in mind.
Yeah, that's irreducible complexity. That's exactly what that is.
yeah, so I get all that, but wouldn't that just be a philosophical bias in the opposite way? So they're saying there has to
no,
be a naturalistic explanation, and you guys are saying there can't be a naturalistic explanation.
I didn't say there can't be. There could be, but when you actually research it, you're seeing the issues and finding one. I'm not saying, oh, it can't be because I believe in God. No, I'm just looking at the data for what it is.
Okay, so from your perspective, there is a possibility that there is a naturalistic explanation, and that it doesn't require an intelligent, like metaphysical designer.
Yeah, if there was, it'd be really impressive. But yeah, there could be, right? There's theoretically should be, but this is the issues that we're seeing when we're looking into the research. I'm not saying it can't be just because philosophically it can't be. No, I'm saying I'm looking at the evidence, and that doesn't seem to be the case.
Philosophically, can't be. But yeah,
I'm
but
I
not
that's
saying
think
it.
I'm different than Taylor on this. I philosophically say there can't be a naturalistic explanation. It
But
just doesn't make sense. There's too many factors.
But then they're gonna say personal incredulity. They're gonna say that basically I'm coming to this conclusion based on philosophy, and I'm not. I'm coming to it based on science.
I know you're
It's not
coming to
even.
it from a scientific basis, which is far more interesting. But I'm just saying, when you look at the complexities of like the cell, it's insane. Like how it just
more
came to
more
being.
solid. More solid than science is philosophy is more solid and more solidified and has proofs in it. Where science, could be debated that it doesn't have proof. So if you have a philosophical reasoning on why it could not be the case, that it would be stronger than a scientific proof.
I mean,
Yeah, dude.
it's the. Oh, sorry. Go ahead, Oscar.
But I was just going to say, I agree with that. So, like, if you just have a philosophical basis of why, like, there has to be an intelligent designer, I think that that's, like, a little bit more solid, in my opinion, than saying, like, oh, no, this is all scientifically based. But, like, I'm going to argue against the scientific basis of something that's only been around 50 years as they try to theorize something that happened 4 billion years ago. Of course, the science would be extremely shaky. But I don't know the science enough to, like, really speak about that intelligently at all. But that would just be the way I'd look at it.
Same
Well,
thing for okay, so if then why don't you get on their butt because they're making a claim that this is something that's feasible and they teach it as if it's verbatim and possible. Like, so get on them. They're doing the
Yeah.
same thing.
Exactly. Yeah.
Well,
At
yeah,
least,
for sure.
at least we're honest with our position, right? These people are basically making, clickbaiting articles about like we created life in a lab, and that's Taylor's point. Is like they're basically misleading the masses, which is not good. It's like the
Yeah.
same kind of misleading. Oh my bad. It's like the same kind of misleading articles of like we can achieve fusion and shit like that. And it's like no, they're way far off from that.
Yeah, people always like proclaim their opinions as like absolute fact, but then when you like press them on the issue, they will admit that it's just a theory or just a fact. And that's what I've heard from the evolutionists is they say it's just a theory. Like we don't have it fully fleshed out. Like yeah, like
Can you tell me
PBS
what you
show.
think a scientific theory is?
But did somebody say something?
What's the definition of a scientific theory?
I have no idea.
Okay, well, a scientific theory doesn't mean the same thing as like a theory of an event that will happen based off of things, like for example, a game theory or something, you know what I mean? Like speculating about a story or something. A scientific theory is essentially something
In
that's
regards to time,
what was that?
a theory in regards to time.
Well, just like a theory used in like everyday
Oh,
life.
Well, see the big
All right.
issue. No, how do you say your name? Gnome's body. Nom's
Sure.
body. Well, the issue though is that this is being taught to kids as if it's true.
Well, a scientific theory isn't the same thing as like the normal definition of a theory. A scientific theory is something that's well established to be the case.
In which it's
Maybe
not.
that's a,
Or they shouldn't say that. Abiogenesis is not a theory.
Is that your claim
that's
though?
a,
Is that abiogenesis is a viable theory, Gnome's body? Is
Oh, I
that your
thought you were
claim,
talking
or
about evolution. I don't know if abiogenesis is a theory, but it would make sense for life to begin. As it, I
That's why I
mean,
say undirected abiogenesis.
maybe I'll ask them next time, like the
well, do
evolutionists
you
or
think
whoever. Like, which
directed
theory of time do
abiogenesis
they
is a theory?
subscribe to? They
No, I
subscribe to
don't.
the A or B theory. I think it would fall in the A theory, right? Of time.
I don't think there's a concrete theory of abiogenesis. There's speculation and hypotheses, but there's not a theory.
Good. We
We can
shouldn't
agree
be.
on that. Yeah.
Has no evidence.
Same thing with
I mean, what
evolution,
would
bro. I
evidence
mean,
even look like?
Evidence would look like being able to recreate these biomolecules at the very least
I mean,
in the same conditions or in a viable condition on prebiotic Earth.
bro, you
Give
just sound
some
like
sort. They don't even have
dinosaur
like a
right
story
now.
of how it could have
well,
happened.
see, here's the thing, Nom's body. So it goes hand in hand, right? Because of the evolutionary paradigm right? That Earth was formed over billions of years. They
Mhm.
need to include a theory about the origin of life. This is where abiogenesis came from, right? It's supposed to coincide with the idea that Earth formed over billions of years, and then all of a sudden life sprout out of it just by some chance or miracle, right? And that one miracle is what they're trying to scientifically quantify, but they can't because they can't even recreate the prebiotic conditions in a lab and go from step to step to step. It's just basically all these assumptions and different steps that they're basically invoking, which is kind of contradictory to their point, because it's basically saying intelligent design is the reason why we're able to do this in a lab. So
Well,
it's just a false premise to begin with.
some things have been created under prebiotically viable conditions. If you watch the debate between James Torr and Professor Dave will go over a good number of them in studies he links.
Yeah, we have done step-by-step census of different things. However, they're in different conditions,
No, they're
and then getting them to come
prebiotically
together and work
viable
together.
conditions.
Do you know what prebiotically viable conditions mean? Do you know how many
Yes,
different temperatures and
obviously.
okay, what pH is that?
pH? I don't know the exact pH.
Okay,
Why
they
should I
use
know the
different
exact
pHs
pH?
and different temperatures and different precursor molecules, by the way.
No, that's not prebiotically viable.
Tell them that. Tell them that. Go tell them. I'd love to hear it.
Oh yeah, why don't
They
you
don't
go
they
email
act
one
like
of
it
them
is.
right now, tell them their research is wrong.
She
I
would
didn't
love
say
to
it's
do that.
wrong. I said it's not cohesive to explain origin of life. I
No, they're
believe their
not
results are what
claiming
they
to
say they are.
automatically explain all of origin of
Not
life. They're just
relevant though.
a single
Oh yeah, well I know
scientific
that they're
paper
not.
regarding
Wait.
organic molecules from prebiotically viable conditions is not a claim to have the origin of life.
But dude, didn't you just say
I
you
don't know.
don't know if abiogenesis is a viable theory? That's what her position is.
Well, no, she's saying that some people's research is wrong.
No, I didn't. I never
You're
said
saying,
that. I don't believe that either. I think their research is right. But you look in the material methods and try to find a cohesive story, and to act like there is a cohesive or viable story is the wrong part. I'm
well,
saying their research is accurate and it's right. And using their results, we can come to a conclusion of, at the very least, the different conditions that would need to be there for each individual part. And we're not even getting to the part where they have to come together and cohesively work.
well,
But
I was
I
I
never
was
said their
saying that
research is wrong.
there have been some studies or like environments created that have created some organic molecules and prebiotically viable.
Yeah, but
Okay.
they're doing a stepwise process. It's not like they're making a biomolecule from inorganic compounds, which would have to happen on early Earth.
Well, yes, they are.
Okay, give me an example.
Might take me a minute to find them. I don't
So,
know
Prove it.
them off the top of my
dude,
head.
in
Take
layman's
as long as
terms,
you need.
in layman's terms, they're basically taking different puzzles, right? And getting pieces from all of these different puzzles, but when you try and put them together, they don't fit. That's the point. There's no cohesive story, like timeline, like, okay, it went from this step to this step during this time period and went from this step to this step, because all the conditions are different for each of those steps to be completed, which is contradictory to the preceding step.
Well, yeah,
That's
obviously you can't know exactly all of the steps that happen for it to occur, but the fact that life does exist is proof that life was
circular
created.
reasoning, bro.
No, it's not.
Life exists, so it must have had to come about by what? By Are you assuming it had to come about by undirected processes? Wouldn't that be
No,
sort of a
no.
philosophical fallacy?
Dude, how do you know life didn't come about like 1 week ago? How do you know?
Oh, haha,
Last
problem with
Tuesdayism.
empiricism. Very, very funny.
I want
I mean,
that paper that
how do
you're
you know?
talking about, though.
How do you know it didn't come about 6,000 years ago as a complete, like, as a completed? If you think of the story of Adam and Eve, I mean, the guy was born. He came into existence. Right? I know you don't subscribe to it, but he came into existence as a full-grown man. I mean, if some scientists went and checked him out, they'd be like, "Oh yeah, you've been around for 30 years or whatever." How do you know?
Yeah, I know. That's just not something you can prove is or is not the case.
We can't prove abiogenesis is the case. Undirected. We can't prove that.
No, but we can hypothesize what caused it.
Well, we can also, I mean, theoretically, then people that are theists could also hypothesize about how life was created, couldn't
You're
they?
Yeah,
welcome to
it'd be the
do
same.
so, but that's not scientific.
Oh, what is science
Because
in your opinion?
like every science, every theory breaks down at
I want to know
like
his
a
definition
triplicate
of
manner.
science, by the way.
Yeah, give your definition, but it all breaks down like a triplicate manner
Science
that you
is
require, like, well, in a second, that you need like three primary things for every single system, and you need them all simultaneously. So I mean, that's like a conundrum in a way. But yeah,
What is
what's
science?
the science?
science is knowledge acquired using a scientific method.
Oh, well, then we totally discount abiogenesis. We have not proven abiogenesis using the scientific method whatsoever. Would you agree with that?
Yeah,
Okay, good.
but
We agree.
life exists, so something caused life to exist.
No shit.
Something did cause life to exist. Is it viable that it happened by an undirected process or just by accident, shall I say? Is accidental
It's
origin of life
possible.
viable scientifically?
It's
That's the question.
possible. I'm not gonna say it
Then
is or
give
is
it
not the
give a
case.
reasoning as to how. Theories should be cohesive and have a cohesive explanation, because theories are meant to explain natural phenomena. So if we don't have any sort of cohesive explanation, then it's not viable.
I didn't say it was a theory. A theory is something that's absolutely proven to be the
No, theories are not absolute. Nothing can be
yes,
proven
they
in
are.
science. Nothing can be
Oh man,
proven. Things can be supported. I'm just letting
well,
you know this. If you take like a scientific terminology course, I can't remember what mine was called in undergrad, but basically it was a course basically learning about scientific terminology and research and scientific method. And we heavily learned that there was nothing can be proven, things can be supported. We can find supporting evidence for it. We can, like,
yeah.
making links to things, like attributing causation to what is it called?
Correlation.
Oh, yeah. Correlation and causation. Yeah. I mean, we can see things
Yeah,
like that, but
if
you can't
nobody,
prove
if you just
anything, by
give
the
up,
way.
if you give up, just say you give up. Like when you say shit like that, just like it just says to me, I give up. Like when you say things like theories are definitions are facts. I mean, it's just
Yeah,
I
that's just
don't
not
say you
true
see
give
what
up.
either.
was I don't see what was resignatory about that statement, but
Theories are
The
not
theory,
facts, quote
yeah.
unquote. They're explanations for
I
natural
never
phenomena
I
that
think
have.
I ever said
Wait,
that they were
wait,
close.
chill. They're explanations for natural phenomena that have evidence supporting them. So you need supporting evidence for a theory, and the
Yeah.
theories are just meant to explain naturalistic phenomena. However, as you mentioned earlier, and we agree on, abiogenesis is not a theory.
Can I ask one question and then you guys can go back to gnomes? My last question is: do you guys put evolution and abiogenesis in the same category as equally unviable, or abiogenesis is less viable than than evolution?
Okay, I guess I'll answer that question, but so it depends what you mean by evolution. Because I don't dispute adaptations. That makes sense. If you have multiple different organisms that have different traits, different alleles, and then one of them can survive, the other one dies out and doesn't get to reproduce, you're gonna carry on those dominant traits, and you're going to have changes like we see with the dogs. We can see wolves, turning into dogs based on mating type, the types of mating that they're using and reproducing with. So that's not disputable. But what is questionable, that doesn't have much evidence, is the in terms of evolution. If I know you're trying to bring it there, obviously, but the generation of new complex specific information that is not from pre-existing sequences is the big question. So for natural selection to happen, you have to have things to select from. So we're assuming that it's already there. That makes sense, natural selection. But where did this new novel information, that's also called orphan genes in the phylogenetic tree, where did that come from? Can we demonstrate this new complex specific information just from random nucleotide sequences? I mean, that's the harder part to demonstrate. But I would not put that in the same category as abiogenesis, which is completely different, because abiogenesis is from non-life, evolution is from things already living.
That was kind of over my head, but it sounds like you're saying like it's at least somewhat plausible that like we evolved from monkeys. I know that you don't believe that, but that would be somewhat plausible. But that life formed from non-life would be absolutely impossible. Is that a fair assessment?
Mm, not necessarily, but yeah. I mean, somewhat. We can get into the nuances of them, but yeah, I would say abiogenesis is much less plausible than quote unquote evolution, and I do acknowledge types of evolution is true. We can clearly demonstrate them using the scientific method. If you reproduce these two animals versus these two, like, or one of them dies off, you have three animals, one of them dies off, those two that are there are going to be able to carry on that genetic trait, and then they're going to be carried on. And you can see this like with hair and eye type and stuff in humans. I mean, it's not really disputed. But yeah, I would say abiogenesis is a whole another category that has like no
Yeah, the problem
cohesive
is
even.
combining them, right? Because what you just
Yeah.
described is selective breeding, right? And there's another field called epigenetics. It could be
Yeah,
totally a
epigenetics is
viable.
turning
Yeah. Sorry.
epigenetics is turning on and off sequences. I'm sorry to interrupt. I was just gonna say epigenetics is a little bit different in that it's turning that is like something adapting to its environment using DNA that's already there. So basically, we can see things adapt to their environment just by turning on and off, or like expressing different sequences of DNA. A lot of things, actually, a lot of these changes that we're seeing are epigenetic changes, where they already have the genetic code and information there to do it. They just go and, like, for example, the histone, which the DNA is wrapped around, will be acetylated. That would cause the histone to loosen, and then that DNA is now exposed and can be transcribed and translated. So like shutting on and off different parts of DNA is epigenetics, and that can actually be carried down between generations too. These epigenetic changes, but see the epigenetics, like I said, it doesn't explain like So this can happen, but it's nothing. There's no new complex specific information. They're just turning on and off genes and information that's already there.
That's my position on it, is that we already have all the things required, right? And it's just a matter of adapting to the environment. That's my position on it. I mean, you could just look at people adapting to their environment in extreme conditions in today's day and age, like Wim Hof, for example, the Iceman. Like, you can for sure see him basically training his body to be more adaptable to the cold versus the other person who is, not in that environment. Like, if you just took some random fat slob on the couch and just put him in on top of the Mount Everest, all of a sudden, like, he'd probably die.
Giga called
Yeah.
out.
Yeah. I mean, that's the thing.
So,
I
like in
don't.
simplistic terms, sorry, just real quick, Beast.
Yeah,
Like
go ahead.
in simplistic terms, and this is like probably a stupid comment to make, but like, do you think it's like at all possible if there was millions of years that monkeys or whatever the hell they say we came from could have evolved into humans?
No.
But why not?
Well, just logically speaking, why are monkeys still around?
Well,
That's not
sorry,
even the
may I
claim they make
interject,
anyways.
please? May I interject? The claim isn't that we actually come from monkeys, it's just that we share a common ancestor with monkeys.
Correct.
Yeah, and that common ancestor is Lucy. That's,
I mean, that's
Uh,
one of them.
Well,
Here we
What is
go
the common
again.
a couple
ancestor, by the
We
steps
way?
did
back.
we did this yesterday with Coach Show, and then we kept taking it back before Lucy,
And
then what it was, and then we going back until he went and said there was definitely went from becoming a reptile to a mammal. So
like
we never
Lucy
repeat
isn't
arguments in here,
obvious.
Elbow. What are you talking about? We never repeat arguments. That's never happened. Come
Do
on.
you mean
It's
Lucy or
novel every
Luca?
day.
yeah, because
Sorry.
then I ask you, what came before Lucy?
Do you mean Lucy or Luca?
Whichever, doesn't matter.
Yo, how about this? Yo, Lefty. Lefty came before Lucy.
Oh, shoot.
Mhm.
Lucy was the monkey that they found that they say was from humans, right? And then Luca was the last universal common ancestor between all species.
Yeah.
Okay, that's what I
And then
thought.
what came before that? Then they're gonna take it to that.
The chemicals dancing around the pond getting struck by lightning, amongst other things.
After that was Righty and Tidy.
Yeah, the only reason why I asked that, and like, I don't have really an opinion on evolution. I've always kind of just not believed in it and had no thought on abiogenesis whatsoever. But I remember seeing like a chart in college that showed like the quote unquote like evolution of skulls that led up to humanity. And I just remember thinking, oh, that actually kind of looks like that's what happened. You know, they slowly got bigger, their face like changed a little bit. Like it looked like, you know, transmuting or transmorphing to from something else to human just based on the skull. So that's why I asked.
Yeah, I mean, there's a pretty solid fossil record. They love to point to Lucy as if it's the only fossil we
really.
have when we have a ton.
Solid.
Yeah, not really, dude.
Are you talking
Like,
to me?
Yeah, not really.
Not really.
Hundreds of missing links in the evolutionary chain.
You know what I noticed about
It
Lucy as
doesn't need to
well?
be complete. Like, it doesn't need to be completely one generation to the next, to the next, to the next. We can just
I
see,
was solid.
oh, it goes from this way, and at one point it went from that to that. It doesn't need to
Wouldn't
be
that make it
absolute.
solid though, as you stated? Wouldn't like these links that show things turning from one thing to another, wouldn't that actually make it solid instead of like making like a post-diction about different organisms?
Make it what,
Say, yeah,
sorry?
everything came from this and
Post-diction.
then yeah.
Oh, it's
I,
your
I
claim. You
think we're
said
I think
solid,
we're
right?
part I
Yeah.
think we're part pigs since that's our closest match DNA.
I think our closest
Okay,
match
so I
to
just try to
DNA is
find
orangutans.
the last common ancestor between humans and monkeys. Let's see. They're still disputing over what it was.
It's some sort of
13
great
months
ape.
ago.
Isn't Lucy based off of like 3
They
random
don't even
bones that
know.
don't
They
really
don't
fit?
even know what the last common ancestor between us and monkeys were.
What about
Uh,
bananas? Don't we share DNA with bananas?
We do, yeah.
So I guess we're part banana.
Yeah.
It's just
I mean,
I
ridiculous.
mean, we're both. Wait, let's see. Are we both eukaryotes?
yeah, what if like some people are from monkeys, but some people aren't?
I'm just
I say
confused.
white people came out of bananas.
I'm just confused as to how this guy just said it was
I mean,
solid.
hey,
And I look up and try to find what is the last ancestor in common between humans and monkeys? Because you just said yeah, we didn't come from monkeys, there was a common ancestor. So I'm like, okay, what is that ancestor? If it was solid, I was expecting to at least see some like genus and species name of something with like a fossil of it. But no. They can't even figure out what is the last ancestor between humans and monkeys.
It's solid.
It's thought to have resembling traits between both species, but lacks direct fossil evidence. How is this solid? I didn't even I don't get it.
Because there's life, Taylor.
Oh,
Yeah, there's
okay.
life, Taylor.
oh
So
yeah,
obviously
exactly
we
what I
came from
said. Oh yeah,
something.
you guys are representing me in such good faith.
That's literally what you
Dude,
said, dude.
what is
It is
it?
not what I said.
You said there's life, so we must have come from something. That's what
I
you said.
said there's life, so something must have created life.
Okay,
Okay.
same thing,
That
dude.
is not the same thing.
He's like,
Okay,
yo,
so Gnomes
he's
buddy,
like,
if that statement "there's life," that statement you made that starts with "there's life," right? If that's not relevant to the discussion, then why did you say it?
He was the one who brought it up.
No,
I
you're the
was
one who said it. So if
Yeah,
it's
you're the
not
one who said
relevant
it.
to the discussion, why did you say it?
Wait, what do you mean?
You're acting as if what you said wasn't relative to the discussion.
I was?
Yeah, you said there's life, so something must have brought it during a discussion about abiogenesis, and then when
Yes,
people try to
because
tie it to abiogenesis, you say, "Oh, you guys are twisting my words." But if it wasn't relevant to the discussion, why would you say it?
we
So
were
No,
it's
I
like,
talking.
asked you for evidence.
Sorry,
isn't it
we were
natural for
talking
people
about
to
common
infer that it's
ancestry
relative?
right now.
Isn't it natural for
it's
people to
okay,
infer
let
that
it
it's
ride.
relevant to the discussion?
Let it ride. It
We,
keeps it fun.
dude, we were
Don't
Him for
let
talking
it-
evidence
about
of
common
abiogenesis, and that
ancestry.
was his answer.
Okay,
Oh,
okay,
wait,
let
wait,
me
Yeah,
wait.
it was his
Let
answer.
let him let him
please,
respond, you
please.
guys. Yeah, let him respond.
Okay, we were discussing common ancestry, and then one of you was like, mentioned what I had said about life coming from something. So that's what I was talking about. I was talking about what I had said, not what they thought I had said.
it's just called being witty. That's
Okay,
all.
so it wasn't relative to the discussion. You just wanted to say that thing
No,
that
it's
has nothing to do
because
with this.
somebody was misrepresenting what I said earlier.
Dude, I asked you for evidence of abiogenesis, and you said because there is life. And then remember, I responded and said, "Oh, that's why I say undirected abiogenesis." Remember?
No.
Oh, okay.
Jeez.
As,
Yeah,
as
you
long
can
as
watch the playback. You can watch the playback. You absolutely said that.
As long as
We're
we
talking
keep in
about
mind that
evidence.
the fossil record is solid.
It's not solid.
It's
Yeah,
solid.
what is the common ancestor between humans and monkeys? I didn't even know that we didn't know that. That's
And
new
a
to me.
theory is a proven fact. It's solid, and a theory is a proven fact.
It's just solid, guys.
Solid.
Why?
Yeah, as far as I
It just
remember,
is.
Lucy is just like 3 random small bones, and they're like apparently knee bones or something. Like,
That's solid enough, bro.
Yeah, Gnomes, I don't
And
know
do
if
you
like
think
you're actually
Lucy
here
would
to debate, discuss, or because that's like just 3 baseless claims. It's just getting kind of silly.
could you
Solid.
list 3?
Was there a reason why they chose the name Lucy? Was that some people argue that wasn't for Lucifer, but was there any other reason?
I don't know. Is
Yo, happy
everybody named
Sunday.
Lucy named after Lucifer?
I think Lucy was after the Beatles song for Lucy in the Sky with Diamonds.
Doesn't Lucifer hang out in the sky?
Well, and that was LSD, so
It was
I want
I
to know
don't
what
know what you
solid
should make
evidence.
of that.
It was definitely named after the Beatles song.
Yeah.
So they don't actually have a common ancestor in the fossil record. They just assume it. Is that right?
They're
Taylor?
still deliberating on which one
Um,
it is. They don't have a
But
direct answer. They say,
right.
"Oh, well, they're thought to have this and that." There's some fossil candidates, but they lack direct fossil evidence. Fossil candidates like Sahelanthropus tactus and Origenus tungus are debated as potential representatives of the LCA, last common ancestor between humans and apes.
Sounds
Yeah,
solid.
I mean, I don't think you can really definitively know what the last common ancestor between the two are because, I mean, there are undiscovered fossils, you know?
So it's just a belief.
Isn't it
It's
weird how we discover a lot of fossils
like
of things that
they're
presently
not
exist?
sorry, they're not saying that it absolutely is the case that that is the common ancestor between the two.
This
But
thing is probably the thing, but we haven't found it, seen it, or
sorry,
anything.
please
But
let
it's
me talk.
probably
what
that
about
thing.
what about the fossil record that we do have, where it shows change over time? That where like the horse heads and even the apes that were found in time, layers that developed into different things over time.
What about the intermediate species? What intermediate species changes do we have? We
Well, the
agree that horse
lungfish,
heads change. We know
lung,
that dogs can change, but where's the intermediate species that's half one animal and half the other? Like, something like
uh,
that.
I think lungfish, mud skippers. I think that they might be an example of an
They still
intermediate
exist. They
species
still
alive
exist.
today. Yeah,
Yeah. So what
alive
about the fossil?
today.
We can't use things alive today as say intermediate species.
Well, they spend part of their time in the water and part of their time on the land.
That's not an
Their
answer.
pectoral fins have adapted so that they can propel themselves on the land, and they might be a good
Intermediate
example.
species. That's something that exists today.
You're wanting to see something before your eyes that takes a billion years. It's like that's kind
The
of a
record has a lot of
silly
evidence.
idea.
You just said the fossil record has a lot of evidence, and I'm asking for any intermediate species fossil. I'm sure they actually do have some. I think that there was actually different genetics, like animals before the flood. So I'm not like, but I still want to know what intermediate fossils do we have, period.
But I think to expect to get a day by day over
Any evidence.
over many millions of years. I mean, that's kind of a
No,
big
she's
expectation,
talking
right?
You're
about
the one that
she's
just
talking about
said that they have
skeleton.
evidence for it.
No,
She's
this
talking
is
about
just par
skeleton
for
by
the course.
skeleton by
Left
Yeah,
Lane claims
skeleton.
it's,
something,
it's
someone asks him to back it up, and then he backs off. He does this over and over again.
What's
I
that
don't know
called
why you
again?
guys
One
Like,
more time?
expect anything different. It's crazy.
She's
What's
not
it
most
saying
called
of the
again?
DNA is
Martin
apes.
Bailey?
It's skeleton by
alright, hold on.
skeleton.
What's it
Dude,
called?
most
Galatas? I didn't hear you. What is that called that he just did? I thought it was a Martin Bailey, where basically you take a much easier to prove claim after stating a hard claim.
That is Amon Bailey. That is Left Lane's
Okay,
yeah.
okay.
I'm not. I'm just saying. I'm just
Okay, good.
asking. I didn't make any claim. I didn't take a stance. I just said,
Yes, you
what about
did.
what
Yes, you
about the
did, dude.
fossil records that we do have showing change over time? That's what I
Where's the
said.
change? Where's the
Okay, Left Lane,
change?
you've realized that when you
Not the
say, what
touchdown.
about this thing that exists, you are making a claim that something exists, right?
Yeah, the fossil record exists,
Okay,
obviously.
so when you say, I didn't make a claim, you understand that you did make a claim.
Well, you can. I mean, everybody knows there's a fossil record. What are you talking about?
Okay, so when someone asks you what is the evidence for your claim, and then you back off and say, "Oh, well, I didn't say there was any evidence
The
for it."
fossil record.
Okay,
How did
what's
you
What
the
is
expect
that?
evidence
evidence
for
for
that?
billions of years? That's what you
right.
Do
said.
you want me to punch it in Google and show you a picture of it, or can you do that?
Yeah, you
It's
can go ahead
your
and do that.
our claim isn't that there is not
There's no progress to be made here. I'm back out.
Our claim
Thank
isn't
you.
that there is not a
He's
fossil
so real
record.
for
The
that
claim
one.
was about how solid or comprehensive it was. I don't know if you just weren't paying attention, but you can't just chime into a discussion and say, duh, the fossil record,
No,
and
I'm,
think that
I'm
you
out. I'm
contributed
out. Just
something.
y'all go ahead. I'm out. Don't talk to
Don't
me
interrupt
anymore.
me, Laflane. You didn't contribute. You just came in to shit the bed again. That's all you
Yeah, don't
do.
talk to me anymore. Shut up.
Yeah, just stop
So,
lying,
I
bud.
mean, I'm not talking
Stop lying.
about left lane right here, right? But it would seem that the atheists like sort of what are they? We can't show you 1,000,000 years. Why would you why would you expect that? And then I mean, the creation of man.
Well, I mean, because
We
it's
can't show
proving
you
their
God,
claim.
bro. We can only show you the handiwork. Okay, so we're all sort of sitting in the same pond, but you like I don't know. The atheists think that they're somewhere in a higher pond somewhere else.
Apparently, I'm the only atheist that won't argue for evolution.
Do you not believe in it?
Uh, at most, I'd say what he said, which is fossil evidence, but again, that doesn't prove evolution. So I don't agree with that at all.
Then how do you think life came about?
Thank you, sir.
Wait,
Reasonable.
the origins of life have nothing. Well, again, this is the fifth day we're talking about this, but some people seem to be confusing the origin of life and evolution. Those are they don't have to be tied together
What
necessarily.
is the evidence for evolution in the fossil record? What are the intermediate species?
Well, he just
I
said that
asked
he
them the
doesn't
same questions.
subscribe to
None of
that.
them have that answer.
Anchovy
I don't.
just said he doesn't subscribe to it. I just want to say, like, he's not defending that claim. He's saying it's absurd.
Yeah,
So
I've
he's
been
like,
Right.
I've been asking them for proof of that for 5 days now, and none of them have it.
yep.
Well, you're
Ooh,
probably asking the
ouch.
wrong
Intermediate
people.
between what and what? Like,
You're the one that
Well,
apparently said they're solid.
let me.
If it's solid, you
Um,
should have a bunch of
Oh,
examples.
wait. Let's let Anchovy, because he's been in this for 5 days now. So, what questions have you asked and who?
uh, I don't remember the other guys' names, but the main one was Tuakna. He's kind of new to the room. I don't know if everybody met him. He's this young kid that came in here talking about evolution.
I
But
remember.
we basically went on a long tangent. He wanted to use fish as an example, and he pointed me to a specific fish. I'd have to look at my internet history to see which one, but he was basically trying to make the point that this specific fish uh, basically evolved into 9 different species that we know of. And I told him to give me evidence of, can you give me any evidence of that common ancestor being in 2 different regions? Either their fossils found in 2 different regions, and if they're still alive, can you give me any evidence of them birthing 9 different species? And always the answer is that all they have is DNA and they don't need any other evidence. So yeah, you're going to be finding a lot of cases where they never find a missing link, and or they never have proof of that missing link birthing different species.
I
But
was
the
there.
missing link cannot be an organism that's still alive. That would just be evidence that this species existed then too, and it didn't change. That would actually be contradictory to their point. So they need to point you to
That's
something
my
that does not
example.
exist.
Yeah, that's what I've been saying to him. So I posted a picture here where he said we have records of fossils, which is like, okay, even if you found those skeletons, now you need to find supporting evidence, which is, can you find that common ancestor in completely different regions, and can you find evidence of them being parts of families that birth different species from themselves? And that's never the case.
I remember Anchovy. I think you accurately represented that convo. I was there listening. Thank you.
Every time I've engaged with Anchovy, he's been one of the most honest and good faith conversationalists I've ever met. Love you,
Well,
Anchovy.
because what happens is, especially with evolution, is they use these terms. They use classifications, right? And when you jump into these classifications, I'm gonna try and post the link he sent me the other day. But when you look at any of these family names, what happens is you find out they're never pointing to a specific species, right? They're pointing to, they'll say, look, it came from this group here, and then you look at that group, and there's over 4,000 species within it, and it's like, okay, so you can, they can never point to a specific species evolving at any point. They just kind of say, oh, the DNA is kind of similar, so this is where we group them.
Well, good on you for asking that. And just like, yeah, where are these intermediate fossils? Like, where is it transitioning to something that we do not have today? That's such a good point.
So wait, question. When if you're trying to get an intermediate species or intermediate like you have to have the fossil, because if they're still among us, then they wouldn't be considered an intermediate species.
Yeah, that's just an animal.
Right.
Yeah, that's
Well,
just
there's
like a
even
lungfish. You know?
I believe I could be mistaken. I think some of these that they say we came from might still exist, especially like with ocean animals. But the problem is, again, they never point to them being in different regions and those fish spreading into different species. So they can claim it all they want, but it's never been observed.
So it's solid.
Yeah, I like to use the lion and the tiger example because people can grasp those animals pretty easily, which is like, okay, if those had a common ancestor, why is it, if they live in different regions, why is it there's no common ancestors found in the
I
two
mean,
separate regions?
sorry, I mean, lions and tigers can reproduce together.
Yeah,
Yeah,
well,
make
they're
migers.
part of the same family, dude. That doesn't say anything.
Um,
There's a lot of animals in the animal kingdom that can reproduce with the same family.
Horses and
That's just,
mules can as well.
horses
yeah.
and
Horses and
mules.
donkeys. Not horses and mules. Misspoke.
Okay.
And a lot of the evidence that I see is microevolution changes, like a giraffe with a shorter neck or a longer neck. Well, we don't dispute that can happen. Things can change traits over time. Dogs, wolves and dogs and poodles, like if you breed them selectively, it's like you'll do the selective breeding, but that's not changing. And actually, the wolves and dogs and all dogs are in the same genus and species. Can you believe that? Same genus. We're talking about subspecies here between dogs, but nobody detests that can exist. So whenever you use an example of like a giraffe with a shorter neck, I mean that doesn't really apply. You need to do some organism that does not exist that shows this change over time happening. Like that doesn't, that was an intermediate species.
Yeah, I'm glad you pointed that out because they get kind of dumbfounded when you agree that speciation exists and that we can observe it because they think that if you're fighting evolution that apparently you're also fighting against speciation, which they're not the same thing. We agree those small changes can happen across time in a certain species, but what we don't agree with is that if you go up the chain in the family tree, that species is going to become something completely different.
Right. That's the crux of it.
Yeah, like genus, like a different genus, something like they have like the order, class, phylum, genus,
Yeah.
Yeah. If you
kingdom,
argue
phylum,
any
class, order,
evolution
family.
defender in here, they're going to use speciation as an example, and it doesn't work.
Yeah, but the speciation doesn't mean they become something completely different, like dogs becoming cats. They're always cats if they're cats.
Well,
And
that's our
humans,
point.
human speciation, they would still be humans. They would be a different species of human. But it's not like that. If fucking we came from monkeys and things like that. Y'all are like misrepresenting when you say that.
No, you're misrepresenting. Instead of using the term humans, you need to look at the
It's
classifications of what you're supporting.
no,
No, but
you're misrepresenting our argument. Evolution
No, I'm not.
is a study that says a certain thing, and when you ask for things to be shown that aren't part of what it says, then you're straw manning the argument.
Hey, I thought you were
No,
gonna
you
back
again.
off, Left Lane. What happened?
Look,
Yeah,
look at the picture I
important.
put in the chat. Right, you're doing the thing I said that you guys do, which is you use a broad term like humans, right? Which is it's a huge vague term that encompasses a lot of different things. And what you're not doing is connecting those different species together. You're just claiming that they came from each other.
So
So you can
to
use the term
clarify
I'm
humans.
not sure what you said there.
You need to be
something,
more specific with your terms because if you say Homo sapiens, that's completely different than saying humans.
wait, why is that different?
Because you need to be specific about which species came from which species. You can't just use a broad term that
Well,
includes multiple species
what's
and
the
claim one
difference
came from the
between
other.
humans and Homo sapiens?
Well, if you look at the picture I put, which is the model he's defending, right? It's a bunch of skulls. There's a tree there they've formed. If you say humans, all those blue ones are considered humans, right? Now, The crux is you need to prove that each of those came from the one previous, and they're not doing that. You can't just group them all together and say this
What
is a
would that look
human.
like, though? I mean, what? I mean, how would you do that? How would you even? I mean, all you can do is take the fossil record and you see the timeline and the changes over time.
Well,
I don't,
again,
I
you would
don't,
find
I don't
supporting
see
evidence
any
with the
way.
fossils.
I don't see any way that you could illustrate what you're saying. I think that would be
Field
an impossible
incredulity.
task.
I've pointed out two different ways. You would need to find these things in the fossil evidence. You would need to find that either one, they were a family together, or two, that the same common ancestor is found in two completely separate regions.
Anchovy, how come I can't just say the fossil record and you're rendered wrong?
Hey, so anchovy, is it something fundamentally different to say like a wolf turned into a chihuahua than to say like Homo erectus turned into a Homo sapien? Are those like fundamentally different in some way?
Yes, because you need to use their specific scientific species name, because what happens is what science does is they group a bunch of them into a very vague term, and then from there they say this came from this. But if you ever look into any of those terms, sometimes they'll
Who
have
does that?
thousands of species within the same category.
But aren't the
Like,
categories like somewhat arbitrary? You know, like someone just said, okay, we're going to draw the line here,
well,
right?
not exactly.
Well, it's
Eukaryotes,
DNA based.
vertebrates, mammals, amphibians.
Yeah, like um there's no explanation in a creationist model of life on Earth as to why there would even be categories that group in this way of like subgroups inside larger groups
It would
to
not
begin with.
contradict with creation, though.
Not necessarily contradict, but it just there's no particular reason why life would have to be organized in this way. If like every organism had just been independently created, you could have mixed and matched traits freely across all species.
Are you saying that you don't
What's the
understand why the
contradiction?
creator would do it this particular way, therefore, like, you don't believe in it? That would be like personal incredulity, right?
Yeah, well,
You
I'm
don't
not
know
even
why.
saying it as a specific counterpoint to creationism. It's just something about reality that the theory of evolution offers some explanation of, but creationism would just be like, okay, this was God's choice, that God happened to decide that there's not going to be any mammals with crab claws.
Yeah,
But
yeah, he made
that had,
everything like he made everything according to its kind.
but
And
So
there's
that's
some
like
things that
why?
also
extremely biblical.
give more
Explanations that
explanation than the design part that evolution can't explain. So it goes both ways.
yeah,
I don't,
explanations
I
are
don't even think it
stories,
goes
dude.
this. I don't think his point holds at all. I don't think it was even an argument there.
What is your argument, ZZ Pop?
Well, I was just pointing out that it's like organisms on Earth are grouped by their DNA in these like very specific nested categories. And we don't see random traits, just kind of hopping around across all of life on Earth. Like there are no mammals with like crab claws. There are no mammals with tentacles. I mean, there's a reason we can even use these words like primate, mammal, reptile, because they're referring to a set of commonalities that can be seen in the DNA. And you can sort of broadly group species into groups and subgroups within those groups and subgroups within those subgroups. So it falls into a type of hierarchy.
Yeah.
And
Isn't
that's
that just
interesting.
classifying and clustering? Like, isn't that
But
normal to do as humans?
well, yeah, we'd look for patterns, but I just mean there was a pattern there to be found. Like it's not arbitrary that, large categories of species all share certain genes in common. I mean, these are real observable things that we can see when you genetically sequence
Right,
animals.
that's still not an argument. You were asking how could a creationist account for
No,
a
it's just
uniform design throughout creation as well as separate kinds? Is that
Well,
your
I mean,
argument that
you
they
can
don't
you
have an
can account
accounting
for
for
it by
that?
saying that was God's choice, of course. I mean, that's always an explanation. So I guess I shouldn't say that there's no way of accounting for it in creationism, but I just yeah, I guess it's a statement of my own surprise then that a creator who could have freely made every species utterly unique would produce such a spectrum of species where so many have so much commonality to others.
Yeah, so that now that you can see the like universal like intricacy and the things that tie together, and see that there was one thing, one being responsible for it. It's easy to see. I hear what you're saying. It's
Yeah,
so
yeah,
easy
it
to
makes
see.
it like it's inevitable. It's almost like it's just like staring you in the face that there was a creator of all this stuff.
Oh yeah,
No,
definitely.
see, I'm drawing
Stacey,
the
real
opposite
quick.
conclusion, because if I was a creator, I'd like be like, oh, let's give some animals that sprout, leaves and
Well, I think we could all
photosynthesize.
thank God this guy ain't the creator, huh?
But wait,
Yeah,
ZZ,
well, I mean, maybe that would be a terrible design, but I'd go
you,
much more wild with it.
you understand it's okay to classify and cluster animals into different species, phylums, kingdoms, etc., just for
Yeah.
organizational purposes. But the problem is making the logical assumption, the logical leap, that because certain things have certain genes, then they must have evolved from one another. That's the problem.
Yeah, I mean, I'm not.
How else would that be the case?
Well, I'm not. I mean, a creator could have just made it that way. So I'm not saying that there isn't an explanation on the other side there. I'm just saying that these categories, it's like they're not just like humans looking at things and being like, oh, I think those two things look similar enough. I mean, that was how it was back in the day, but now we actually have genetic sequencing. So now we can actually say the definition of a mammal is something that has a particular set of genes, and that defines the mammal category.
That's fine. That's
Yeah.
totally fine to do. Again, it's not pertinent to the point though. It's like even what Gnome's body just said, he was just like, well, how else can it be the case? Like to have genetic consistencies throughout different kingdoms and animals and stuff like that. It doesn't mean that it's true, right? Like it doesn't mean that we
Well, yeah,
evolved
but you
from
didn't
a
answer
common
the
ancestor.
question.
What was your
Just to
question?
be clear, before we move
How
on
else
from
could
that,
it
the
be the
reptile.
case?
All right, never mind.
Go ahead, Jeremy, real quick.
No, I just want to be clear. Like you're talking about reptiles and mammals and stuff. Just we ironed this out last night, and all the global evolutionists were agreeing that
Well,
at one point reptiles turned into mammals. Like there was no mammals, just reptiles, and then they evolved into mammals. So that's one creature turning into a completely different creature.
No. See, that
I
you
don't
would
even
just
know
say
that.
mammals are a subtype. That whatever is the formal scientific name for reptiles would then include mammals as a subtype within it. So things are always just a new branch, like off of the branch they came from.
Yeah, but there's a day where you had no cats, and
Yeah.
one creature, this other thing, turned into cats.
Well, but
in a
cats would still
simplified
be
way,
in the
yeah.
category of what that creature is. It's like,
I know you want to play word games, but there were no cats, and this one creature turned into cats.
No, you're just oversimplifying it. We're not playing word
I'm not
games.
oversimplifying it. That's how
You
you
are.
have to have it happening. Otherwise, you don't have any evolution.
no, what we're saying is that cats are, a modification variant of whatever is the thing that, was the ancestor that had preceded them. But like they still inherit all other classifications from wherever they descended from.
Right, but we know that there was an animal that eventually it turned into a cat. It wasn't a cat, and then eventually it turned into a cat. You
The
have to
Adam
have
and
it
Eve.
that way,
Yeah.
otherwise
Yeah.
you don't have evolution. I can't believe evolutionists are fighting against
Well, yeah,
things turning into other things because you literally don't have any evolution without that happening.
yeah.
I think it's a bit of a linguistic thing when we say things turning into other things.
Yeah, it's called word games. The Globers, the
The
evolutionists, you guys love it. Word games, we know.
it's sort of the point being, the evolutionary tree is modeled as a tree. So when we say that like new species arise, it's that there are twigs that grow off from one branch that eventually grow into thicker branches of their own that eventually keep splitting. But it's not like a branch from one part of the tree suddenly like teleports somewhere else to like become its own thing. Like things are always diverging off of wherever they came from. So they're always within the larger category of wherever they came from. They just represent a new subcategory within that. Like every descendant of mammals will always be in the mammal category. They will just represent new subcategories of mammals.
And they'll all be vertebrates, and they'll all be
Yeah.
eukaryotic.
What?
Yeah, that's just what I mean of
eukaryotic is just a type of cell.
When is a dog not a dog? When did a dog change from something else? Because this is like double speaking, guys, right? If
I
your
don't know.
theory starts in a pond,
When
let me
did
finish.
wolves turn into?
I'm
My
not
bad.
done, bro. Not
Sorry.
done. Not done. In your theory of evolution, you start off in a pond with a single cell. So, in order for that to happen, to have evolution, one thing must turn into another thing. There's no way around that. No way.
Are you done?
Yes, I'm done now.
Okay. Apologies for interrupting you. Yeah, I don't think anybody who subscribes to the theory of evolution is claiming that one thing doesn't come from another, because obviously that's essentially just how transformation works, you know? So I see in your profile picture you have a dog, and I guess the question would be, when did wolves turn into dogs, you know? Because dogs are
No.
domesticated.
No, that's different. That's different.
Sorry that's a different question. Sorry to interrupt you, but when was a dog not a dog? Is the question.
Remember what
Right?
Anchovy brought up? How
Well,
that
I
that's
don't see exactly how it's different. Could you tell me why it's different?
You don't see how it's different from a
Like,
single cell.
what? No, sorry. I'm just saying, like, when was a wolf not a wolf when it became a dog?
Is a dog not
Forget
a wolf?
about wolves and dogs, bro.
It's
Forget
still
about wolves.
canine.
Okay, sorry. That was okay. So,
In,
could you
in
just
your
ask?
evolutionary theory, did we start off as a single cell in a pond? Yes or no?
Well, that gets back to
Uh,
abiogenesis, which I mean, we don't
No.
know what.
This is not abiogenesis. This is
that's
after
yeah, that's
abiogenesis.
after Abiogenesis. Because
Well, yeah, but look,
abiogenesis is basically saying non-life getting to that one single cell. So let's just skip that part and go to the one single cell. Go ahead, Essie.
Yeah, it's very simple. You have abiogenesis, no matter how it happened. I'm not saying I believe it. So we have the single cell. Boom. Now, after that, right? Is it possible for you and me to be here without that single cell turning into multiple cells, those multiple cells turning into flagella, whatever, and then that turning into a fish, and then the fish going? Is that possible without that cell evolving and one animal turning into another?
no.
No. So there you go. So your evolutionary theory, right, dies right there, because you cannot show one instance of a transitional fossil. Show me one instance where you have a transitional fossil, where you can show one creature turning into another, now that you admitted it. Can you please show that?
If you want to look up pictures of transition species,
There isn't any.
why don't you check?
I have. I'm
Well,
challenging
every
you.
fossil is interpreted as, I mean, some organism of some species that came before that is some branch on the tree from an earlier point that leads off to species that exist today, you know, as well.
What if they're exactly the same as the species now?
Yeah, you know, you could take like trilobites and stuff. It's like, yeah, I mean, like not every species um, undergoes a lot of change. Some are steady, pretty
Can you show
stable.
us one
They find their
example?
niche. Well, like trilobites. I mean, they're sorry, horseshoe crabs. Um, that's what I was thinking of. Like there's fossils of horseshoe crabs that are dated for super old. I forget exactly how old, but super old fossils of horseshoe crabs that look basically the same as the horseshoe crabs that we have crawling around today. So,
Well, yeah, that's it. That's the worst argument you could possibly give for
the
evolution.
example of something
Blue-blooded.
that found a niche and has been living in that niche for a very long time.
Oh, but wait a second. If you're using that as your example, wouldn't that be the same exact example that Anchovy brought up earlier about
Yeah,
having a
I don't think
short-necked giraffe versus a tall-necked giraffe, and how you can have different traits evolving? Like that would be
he
the
was
same
using
thing.
that as his evidence.
Well
No,
then,
but are you
okay.
guys aware of what animal he's talking about? The blue-blooded creature
Yeah.
that they say is one of the oldest thing. Like that is an example of non-evolution. Like I
Yeah,
don't
I mean,
understand
well,
why you would
things
bring that creature in.
I
well,
mean,
things don't have to mutate. I mean, there doesn't necessarily have to be pressure that um, amplifies, mutations within a species. A species could end up in a very stable, situation where it continues to be successful as is. Changes get weeded out rather than promoted.
So I posted a picture just as like a visual example of what I'm talking about. So um, you got the different species, right? And if you notice, what we're trying to say is where those lines connect, that's what they would call a common ancestor, right? And you'll notice in a lot of these trees, well, obviously they're not going to have a picture because a lot of them, they don't, they've either never found them or they can't imagine what they would look like. But on top of that, even if they claim to have a common ancestor, you need supporting evidence that says this common ancestor eventually lived in two different regions, right? And split into these two different animals. And that's the evidence that we're missing. So even if you find a fossil for some other species that doesn't exist today, you still need to support the idea that animal turned into different species. We don't have that.
I don't see why it needs necessarily that the fossil that's thought to be a common ancestor needs to be in multiple places because animals migrate later on.
Well, they would be because of you understand that okay, so when you make the argument that evolution takes so long that we can't see it, what you're saying there is that over that time span, I should expect to see that common ancestor in two different regions because of how long it took for it to change. So you can say they migrated only once they became a different species, but you also have no evidence of that.
well, how would you prove that though?
Because you're telling me it takes millions of years for this change to occur. We should expect to see that one common ancestor in two different regions that you're claiming its children species came from.
Sorry,
At
you didn't
least.
answer my question. I was ask you how would you even prove that was the case though?
You would find
Again, fossils.
Yeah. Yeah, you would find a fossil in two different regions. There you go.
So, well, are you saying that you would find like different fossils along the path of migration?
No. Well, I mean, sure,
It
maybe, but
depends. It depends on the animal. Yeah.
it just but there's no fossil record whatsoever of that intermediate species. That's the crux of the argument, though. And not only that, he's adding to it saying that not only would you have to find it, but you would also have to find it in different regions. Am I
I would
steel manning you
expect to
right?
find.
Yeah.
Yeah, that's correct. I would expect to find a common ancestor's skeleton among other skeletons of species that further came from it, right? Because at some point it would have, when it split into the different species, it would have lived among those species.
Mm-hmm.
Yeah, and I've yet to see like how do they explain the transition from sea to land, right? It's another tall tale. Like how does that
Here comes Left
how do
Wing with the
You
mud
wait,
horse.
lungs
you
from
say you
gills
would
to lungs like
Called it.
you wait. You said you would expect to see the parent species among the one you're looking at.
What?
Dude, I swear
You
I
would
called it,
expect
bro.
yeah, because of how long the change set takes that you guys are claiming, you would expect to see skeletons morphing over time, right? Between that species and its parent ancestor.
Yeah, transitional fossil
Well,
things.
we see that though,
But
though. I mean,
no, you
we
don't.
posted images of that. Yeah.
You don't
No,
see
you
that. You
didn't.
find two different skeletons and you claim one came from the other.
And you posted a couple of bones and some drawings added to them. Like,
Well, I mean, it's
yeah,
sort of
come
like,
on.
and I'm not, yeah, and don't get angry. There's no need in getting angry, but I mean, the
I'm
way
not
that I kind
angry.
of,
I'm in a great
no,
mood.
but I'm just, I know you're not, but I mean, what I see is look at the like where they've got the lineage of speciation lined up in the skeletons. You got the first one and the last one, and the ones in between are intermediates. But I feel like I don't know what y'all are asking for would look like. Would it be more similar from one to the other, or what? I don't know.
No, it's an impossibility. What we're asking for cannot exist because it is impossible. Because a species has to be intact. You can't like have half a lung and half a gill. That doesn't happen. That only in fantasy. This transitional things that you guys think are just impossible because an organism needs all its parts in order to function. Evolution is just a fantasy. That's why there's no fossils for it, because it just can't happen that way.
Yeah,
Yeah, that's
we'll say improbable. We'll say improbable, but there's no intermediate fossil found. So
yeah,
right,
and I was gonna say, you can make the argument, Left Lane, that you know, every species you're putting in between is the intermediate, and that's easy to say with things that look alike, right? But to their point, if you go up the tree far enough, eventually that thing's not going to look anything like it does right now. So where are those intermediate bones?
Yeah, that's true. That's I think it's unfortunate that we don't have, the fossil record of all of the animals that ever lived. That would be cool, but I don't know.
So, Left Lane, do you agree with the other gentleman that one thing has to turn into another thing? Because you guys were fighting that for I don't know how long. Are we now in agreement that evolution one thing has to turn into another thing, or not, Left Lane?
Well, I don't agree that like, for instance, a dog would turn into a cat or vice versa. I mean, whatever a dog evolves into, it's still a dog. It's just a different species of dog.
You guys hear that? You guys hear that? So maybe like the camp is split.
Well, I mean, and it is sort of great, but I mean, it's like, crocodiles are vertebrates and we are too. Fish are vertebrates and we are too. That kind of thing. We're still vertebrates. We're just a different species of vertebrate.
Yeah, but Left Lane, you're saying a dog is always a dog. Now, the gentleman before and the other guy, we agreed that in order for there to be an evolution to start from a single cell in a pond, one thing has to turn into another. But now you're saying no, a dog is always a dog. A cat is always a cat.
I guess if you look at the whole thing, every animal came from an original animal, right? That single cell organism. But it's such a mind-bending idea to contemplate 3 billion years of these changes going on among the animals that what happens there, but I don't know.
The only thing that's mind bending is that people believe it.
Yeah, I agree, bro. Yeah, you can't
Well,
have
no,
it both ways.
it's not it's for me, it's not a solid belief. It is an idea that it appears that there are things that happen that we can observe today, right before our eyes. And generation times, you got to think, humans, what generation times, like 20 years, but we've got these bacterial species that their generation times like 30 minutes, and we can watch that happen before our eyes. We're never going to see like animals, any complex critter exhibit evolution before our eyes. I don't think.
So it's just a belief.
No, it just requires the concept of
belief.
Many fucking years and many generations.
It requires belief, Left Lane. It's just a belief.
Yeah, and the many years, right? You guys shoot yourself in the foot because you say you have a fossil record and you can layer it down. So we can ask you, okay, if you got this many years and millions of years of record, well, you should have it in the fossil record.
No, it's not a belief, though.
Bro. It's not
yeah,
existent.
I mean, I reiterate, it's not a belief that I hold. It's an idea that is potentially valid. But yeah, I don't think it is a solid thing from my perspective, anyway.
Do you think it's true?
Oh, I don't know.
I mean, Left Lane. So what we mean by things turning into different things, we're not saying it's overnight, but if you look at that picture I just put, and you start at the bottom, and it says that the different bears, a bear and a panda had a common ancestor, and then that common ancestor also split off into seals and walruses. And if you follow that back, that was the common ancestor that then splits into the foxes, and the tree all interconnects. And what we're saying is where those lines connect, there's no evidence for that. It's just posited based off of DNA.
Just want to clarify, not all of us grant the claim that pandas are real.
The thing is, that's a great chart to put there. So where's the common?
It's their own demise, right, bro? They give us their own thing, and it's just not there. I mean, you can't have it both ways. You can't start off in a pond with a single cell and then say, well, one creature doesn't turn into another one, and then say, well, it does, but the genome and this and that. No, you can't play wordsmithing with this. It's either one or the other.
Yeah, and it's the equivalent of like if you were arguing against a Christian creationist, right? You would use the Bible to argue against them, right? It's the same kind of thing. We're using your evolutionary paradigm to argue against you.
Exactly, bro. Exactly.
Well, I mean, my inhibition is purely incredulous that it's kind of mind blowing, but I do offer it validity as an option because of what we do know. How about that?
Your position is entirely incredulous. Thanks.
Not
Lefty.
entirely, not entirely. No, not entirely at all. Don't misrepresent me.
See, this is really easy to argue against, right? Because by your own showings, you show that you start off in a pond, right? Single cell, and then this turns into that, and that turns into this. And it's either you say, well, you don't understand, and it's not like that. And if you say, well, it is, then I can ask you, okay, where's the fossil evidence? Show me a transitional species, and you just can't do it. So, I mean, either way, I mean, this is a ridiculous. We have to understand it's just one dude in the 1800s. He went to an island, he saw some birds and a couple of turtles, and he came up with an idea. That's it. Like there's nothing there. There's nothing there to it. There's no proof at all.
Yeah, I don't think there's any evolution proponents left in the chat. ZZ Pop left, Gnome's Body left, and Left Lane's incredulous.
See, you're going to take what I said and turn it into some kind of negative comment. That's what's wrong with you, dude. You're constantly doing shit like that.
I'm just taking what you said, bro. You said I'm incredulous to this.
That's not what I said. That is not what I fucking said, dude.
Okay, what did you say?
I was very careful to say that I do see things that are practical. That I know that there are situations exist where we see bacteria evolving to be antibiotic resistant. We see I can't remember if it's a mold or a fungus that they actually encapsulated that is actually evolving to be tolerant of different conditions, things like that.
You didn't say that at all.
What I said was incredulous for me, as that it is a mind-blowing idea to think about the single-cell organism turning into what we have today in the animal kingdom. That I claim incredulity because it's mind-blowing to me. That's what I said. I didn't say I'm incredulous and I'm arguing from ignorance. I did not say that. So if you don't understand what I said, then that's on you.
You have an incredulous position. You just said it's mind-blowing to think that a single cell came from
You're going to fucking
billions of
continue
years.
to misrepresent me.
Am I crazy, or did he say that I have an incredulous position because it's so mind-blowing?
If you leave out all the other shit I said, y'all be honest and tell him what he said.
Okay, well, either way, is there any other evolution proponent in here that disagrees with what Anchovy put forth, or SE? And if not, we can just move on to a new topic, bro.
Wait, I had a quick question because you just said something there about funguses and bacteria becoming, antibiotic, immune, or like, So why wouldn't that just signify adaptation and not evolution? Because they're not doing anything outside of their DNA or the cell structure's ability. They're adapting to their environment according to what their cells will let them do. But you're not going to see a fungus turn
No, that
into something else. You know,
no,
like
they do DNA analysis on these. It's actually in a very high population or shit. I lost the damn word. But where they're reproducing rapidly, and then you have like one mutation per million individuals. Well, where you've got millions of individuals reproducing in 30 minutes, you have many mutations, and you can analyze their DNA. You can have the DNA sequence of the ones before and the DNA sequence of the next ones that are resistant, and it's the result of mutation where they're just random mutation has led them to be antibiotic resistant.
well, it wouldn't be random if they're pumping them full of antibiotics. That's an adaptation of them trying to protect their own species. Like that doesn't seem necessary as
Not analogous.
it doesn't seem analogous to like, oh well, it's definitely evolution, therefore we came from primordial jizz.
It's not analogous. It's like
Yeah, you would have to explain the different DNA sequence. Then I don't know how else to explain the DNA sequence changing.
No, can DNA change in a relatively short period of time based on the environmental factors? Yes.
Yeah, we know that because we
no one's
have
arguing against that.
Not necessarily.
yeah.
No,
Yeah, we do
it's way
because
more
it's called.
complicated than that.
But no one's arguing against
Then what
that.
is the vaccine? The vaccine changes your DNA rapidly.
No, it doesn't. No, it doesn't. It does not affect your DNA.
That's incorrect.
It's an mRNA vaccine.
Yeah, it is a
Attaching
messenger.
itself.
It's a messenger ribonucleic acid, which provides the cell the instruction. It provides the instruction for the mechanics to produce this spike protein. It doesn't.
Which part of the cell?
And the reason it does that is because you don't have that in your DNA to make that protein. So it provides the instruction to manufacture the protein.
So it changes your DNA.
No, I did. You see, there you go. No, I didn't want to say it.
It does change your DNA because your DNA was one way, and then you got a vaccine.
No, it don't.
Now
No, it don't.
Now it's another way. And
You don't know what you're talking about.
The spike protein, dude. That's changing your DNA. It's called gene therapy.
That's not how it works, dude. You don't know about protein synthesis.
All right. Well, whatever. This is not analogous to the previous conversation, anyway. You brought up
Proteins
bacteria.
proteins are manufactured according to the instruction of mRNA within your cell. You don't have those mRNA in your cell to make that spike protein. So we put it in your cell so you can make the spike protein. And it's as simple as that, really.
It changes your DNA. Yeah.
No, that ain't what I said. You don't know what you're talking about.
Literally changes your DNA.
Widowy.
Just say it again. Might be true.
It widowy changes your DNA.
Widowily.
What if Elmer Fudd was a walk stall?
He would fuck that wabbit up.
Must I say, Discord is not filled with experts on scientific topics. So,
Are you
I
on?
would. What was that?
Are you one? Are you an expert
No.
on scientific topics?
Oh,
Are
so I
you?
bring that up.
Yeah, thanks.
Are you?
Are you an expert
No,
in?
you're the
in
one bringing it
Discord?
up, though.
No.
So why would you bring it up if you're not one of them?
Sorry, let me finish. Okay.
Okay.
I'm just saying, do not get all of your information from random chaps on the internet. Look into what experts are saying, and look into what the actual critics of the experts are saying. But don't exclusively look at the critics, because a lot of the time they do not present it in a fair light.
Yeah, way to assume, bro.
Assume what?
It's a very strange thing to say.
It is
Wait,
very
are you
weird.
saying there's information outside of Discord?
No way, dude. Really?
No, wait, what? No, I never
Wait, the
said
whole
that.
thing you just said was just poisoning the well on anyone's credibility. Like that's the only goal of what you just said. It doesn't offer
No,
anything. It
no,
doesn't
no.
offer anything to the discussion. It's just, hey, maybe we don't trust what people are saying here. Like, it does. What productive does what you just said offer to the conversation?
Let people know they should research valuable information that comes from experts.
Oh, they didn't know that ahead of time until you said that. I guess they're not that smart.
Thanks
That's
for
kind of
coming
a
in, bro.
wild assumption
Appreciate
to
it.
make.
Yeah.
Thanks, man.
Thank you, bro.
Very
Sorry,
useful.
do
Everything
you-
you've said tonight has just been so useful.
Duly noted.
Yeah, I'm gonna
Thanks.
start doing that from now on.
Sorry. Do you guys look into what experts say?
Oh, and now
Yes.
you want to ask a question instead of assuming. Yeah.
Yeah, we do.
Most people
Could you
here
give me
do.
an
That's how they got
example?
to this conclusion.
An example of what? What experts say
An
on what
expert
topic?
you're looking
Yeah, what
into.
topic?
It doesn't matter.
Yeah, it does matter because you can't just say experts. There's experts on fucking everything.
All right, no, look, we don't need to entertain this, man. You're just trolling. You've been trolling all
I'm
night. You
not
made like 5
trolling.
baseless claims. You're making another baseless claim. I don't know what your point is, what you're doing here.
What claim was I making that was baseless?
That we haven't
So the
looked into it
claim that we
ourselves.
haven't looked into expert opinions,
I
but
never
also
said
that
you didn't look
don't
into it.
interrupt people, man.
You interrupt me all the time.
No, I don't.
Okay. Sure. All right.
Dude, let's just
Anyway.
hold on. Let's just get back to like a more productive conversation. Something that's not
I agree.
personal back and forth. Something that's we could shift to something new if needed. But it seems like it's headed into a personal sword fight.
Stupid.
Tell me I say things I didn't say.
Yeah, we can just move on.